I'm so glad all of you guys could make exciting. Excellent, it's even better. So welcome to Writtle. I'm so glad all of you brilliant people could make it, despite the trials and tribulations of every possible form of transport not working. So well done, you will get a gold star for this. And also, I'm really excited that we've got so many different types of scientists in the room today. I don't think I've ever been to such an interdisciplinary meeting as we have today. And I think that's really, really important and a really nice way to start this special interest group on internet ethics and welfare. You know, we've got representatives from vets, we've got zoos, we have a variety of entomologists from very different backgrounds. So really, really excited to have all of you guys in this room today. And hopefully this is going to be a really lovely, informal, friendly environment for us to chat about some really interesting ideas. So thank you very, very much for all coming today. I'm really excited to hear all of your presentations. Just as a little bit of housekeeping, those of you who are here in person, please have your devices on mute. Questions at the end of talks, well, you can find any of us. For those online, please do keep your cameras and microphones off. You can pop your questions in the chat, and do feel free to share any contact details if you want to get involved with networking as well. So just a big thank you, and over to you to chat about the RES.
Thanks very much, Anna. Hi everyone. What is this clicker? Yeah. Lovely. Hi everyone, I'm Emily. I am Head of Publishing at the Writtle Entomological Society, so welcome from us. I'll keep this quite short so you can get on to the good stuff. Yeah, this is the staff at the RES who are around today, so that's me in the middle there. Amy is our events manager, she's in the corner. Ask her if anything goes wrong, I don't know what I'm doing. And for folks online, ask Bianca if anything goes wrong again, I don't know what I'm doing. Yeah, so that's us, always happy to help. Massive thanks to Eleanor for organising this meeting and to Writtle College for hosting us. We're really excited to be here. And yeah, at this point, I guess really just generally really excited about this first SIG meeting of the Welfare and Ethics Special Interest Group. Really exciting new field that's like rapidly developing. And as Eleanor said, great to have so many different people in the room, and really excited for the RES to be involved at this early stage. And we're really keen to facilitate these conversations, and yeah, great to have you all here, and please do spread the word about the SIG. So just a little bit about the RES. We're a membership organisation, we've got members all over the world. We have a big student contingency in our membership, and membership is free for students in your first year, so if you know any students, if you are a student, please do join us. You get discounts on our events. And access to our grants and awards. I'm Head of Publishing, so I look after our journals and our book series and our library. We've published 7 international scientific journals and a handbook series on insect taxonomy. Our library is fantastic. It's one of the leading entomological libraries in the world, and you can access that from anywhere via our librarian, Rose. And yeah, lots of exciting stuff to get involved with the RES. Upcoming events, so particularly relevant to you guys, we have a rearing SIG on the 6th of July in Norwich. Gary is organising that, and he's in the room here, so please do register if you want any more information about that SIG. Have a chat with Gary. We also have our annual conference, Ento, in September. This is going to be a fantastic meeting across all of entomology. This is in Falmouth. So a beautiful part of the world, and yeah, if you join as a member, you get a very big discount. Early bird deadline is today. Is that true, Amy?
It is 5 p.m.
So yeah, if you want to register at the cheap rate, do it now. Not while people are speaking, but you know, lunchtime. And yeah, lots of other events coming up. Have a look on our website. We've got some committees if you want to get involved more in the society. We've got some vacancies on our committees at the minute. So education and training, which is a brand new committee. Very exciting to be setting that up. And if you want to be in on the ground floor with that, do have a look on our website and get involved. Similarly, science policy and society, a brand new committee for us. Finance and publications too. I look after publications. Publications are great. If you want to get involved, again, have a look on our website. Just wanted to mention a couple of quick things that we've done recently. So Grand Challenges in Entomology is a paper that's come out recently in Insect Conservation and Diversity, one of our journals. This was based on a massive horizon scanning exercise with the RAS membership. It came up with lots of key challenges that need addressing in entomology. And one of the things that came out really strongly in that was interdisciplinarity. And I think this SIG particularly is in a great place to kind of address some of that, and particularly interdisciplinarity across research disciplines, but also engagement with other kind of communities of entomologists, including amateurs and the general public. So I think a really interesting read and a really good kind of place to start if you maybe are new to entomology, so have a look at that paper. We also had a garden at the Chelsea Flower Show last week. I don't know whether you saw it on telly. It was pretty exciting. It was designed by Tom Massey, who's a fantastic landscape designer, and sponsored by Project Giving Back. You can find out more on our website. But I think one of the things that came out of that really strongly was that it feels like the public perception of insects in gardens is changing a little bit. So there was lots of, I guess the crowd at Chelsea has traditionally been quite maybe anti-insects. They want to get rid of pests in their garden. But actually during the week, we really talked a lot about having different habitats for insects in your garden, and people were really engaged with that. So it feels like a great time to engage with the public about entomology and having that insect presence in your local green space. And again, that kind of fits in really well, I think, with what we're talking about here. You can find out much more about the garden and see some really gorgeous pictures on our website, so do have a look. I think that's me. So yeah, enjoy the meeting. I'll hand back to Eleanor to introduce the first.
Great, thank you very much. All right, well, I will put the slides up. Okay, please, yeah, go for it. Come up. So I'm very pleased to announce our plenary. I have some notes here to talk about the wonderful things that she has done. So I'm very pleased to announce our plenary. Dr. Meg Merritt. She is currently a postdoc at California State University, where she's looking at thermal adaptations to solar radiation. However, she is also very involved in insect welfare and has been a real driving force of developing collaborations on welfare, particularly looking at farmed insects. She's published extensively on issues of pain perceptions in invertebrates, and she is also the founding director of the Insect Welfare Research Society. So we're very, very glad to have her here. She's flown out all the way from America, so it's a real treat to have us with her today. So thank you very much.
Thank you so much, Eleanor, for introducing me. I'm so grateful to be here today with all of you. This does feel like a very momentous occasion. I'm going to be talking a little bit about my work on insect welfare and ethics in this emerging field context. And I want to start by thanking all of these folks, some of whom are in the room today, who have really played a vital role in the work that I am talking about today and my own understanding of insect welfare and sentience. So thank you very much to those people, especially those in bold. So this is the outline of where we're going with our talk today. We're going to start with whether or not insects could matter morally, because that's fundamental to the question of whether they have welfare that we should be concerned about at all. After that, we're going to move into a specific use case of insects that I'm very interested in, which is insects used as food and feed, or those that are farmed at scale. So if you're not familiar with that industry, I'll introduce you to it and then talk very briefly about some welfare concerns we might find for black soldier flies. And then finally, I'm going to talk about opportunities for this field moving forward, which I initially framed as challenges, actually very similar to the Grand Challenges paper itself, but I tend to think of challenges in research as opportunities for research, and I think that's the right framing moving forward. So we're starting here. Could insects matter morally? So this is the ethical question at hand. What traits cause us to morally consider the welfare of others? I have two images on this screen, and presumably if I went up to one of them and I gave it a good hard kick, you would be like horrified. And if I walked up to the other one of them and did the same thing, you would be like, nah, whatever. So you must have some trait that you're already using in your mind to decide that the welfare of one of these things matters and the welfare of the other one does not. Presumably that trait is not cuteness or roundness or the presence of eyes, because both of these things have that. So you've probably already intuited what trait it is that you're using, and it's summed up relatively nicely by this quote by Jeremy Bentham. It's, can they suffer? Can they feel pain? We believe the mouse can feel pain, so we should not kick it. We should not cause it harm. We should consider its welfare. We believe the rock can't, so it's fine. So when we think about the ethical use of non-human animals, what often matters is whether or not we think they are likely to be sentient. Now, sentience is this umbrella term that means the capacity to have subjective experiences. One of those possible experiences is pain. There are many others. Because the discussion by entomologists has focused on pain, I'm going to focus on pain today. They're not perfectly synonymous, but for the rest of the talk, we're going to treat them kind of as though they are. And when I say the capacity to feel pain, I mean feel pain. I do not mean nociception. So actual pain. Now, the distinction between these two things may not be clear, so I want to clarify that for all of us moving forward. So nociception is our reflexive responses to noxious stimuli. So imagine you reach your hand out, you touch a hot stove. The sensory cells in your fingertip are going to send a signal to your spinal cord, which is going to send a signal back down to your motor neurons, which are going to pull your hand away reflexively. And that happens without you experiencing any pain yet, that reflexive nocifensive response. Now, pain happens when the spinal cord sends a signal up to your brain, and that is where you feel pain, right? So you're going to have some kind of subjective awareness of that experience. You're going to say, ow, you're going to reach for the burn gel. So these are distinct from each other. One is a subjective experience, one is just a reflexive response. So the challenge with subjective experience, you're probably already aware if you work in science, is that it is an inherently private, non-objective experience, which makes it extremely challenging to empirically measure. So one way that we can try to get at subjective experience is just to ask, hey, how did it feel when you touched that stove? It hurt. Oh, okay. I now recognize you have had a subjective experience. This works very well when you can communicate with the animal in question, but there are, of course, many animals we cannot communicate with in that fashion. And this has led to some things that we might now view as morally not okay. So one example would be human babies, obviously not great at self-reports early on in their developmental period. In the 20th century, we conducted research, or we conducted surgeries on them without anesthetics, because we just assumed that those responses that we were seeing when that surgery was kind of happening was a nocifensive reflex response, not actually the experience of pain. Similarly, we often live dissected dogs in the 17th century, and the scientists that were performing that work suggested that this was just nocifensive reflexes responses when those dogs were responding to that dissection. Most of us today probably aren't jazzed that those things happen, wouldn't want to see them repeated, so we probably should agree on some objective set of criteria we can use, not to prove subjective experience, but to demonstrate when subjective experience is sufficiently likely that we should be cautious in how we treat that group of animals. So that is what we tend to do. We tend to look for major lines of evidence that help us raise our likelihood or probability of certainty that a particular group of animals could be experiencing pain. And there are two major lines of evidence we tend to use: neurobiological evidence, because that is the substrate of the pain experience, and behavioral evidence, because this is the fitness-relevant output of the pain experience for which we think it may have evolved. So this brings us to the animals that we're most interested in here today. What about the insects? What do we have to say about their neurobiology and their behavior? So this has been a question that many entomologists have been interested in throughout the 20th century. This is a Scientific American article by Harold Bastin all the way back in 1927 about this particular question. But the first time somebody really went through and tried to more thoroughly review the evidence is going to be Eisman et al. In 1984 with their classic paper, Can Insects Feel Pain? A Biological View. Now this paper goes through and they look for some neurobiological and some behavioral evidence in relation to this question, and they conclude that from the evidence they have at the time, there is no adaptive role for pain perception in insects. So really quickly, some of the evidence they rely on: insects have no nociceptors. Remember those sensory cells I said in your fingertips that tell you that there's a noxious stimuli that you should do something about? Insects don't have those, so they don't have them, unlikely that they're going to respond and feel pain. They have a simple nervous system organization. Eisman doesn't specify what about the nervous system is too simple, but lots of other entomologists have evolved this argument over time. The most recent version is by Shelley Adamo in 2019. She talks about the lack of direct connections between sensory integrated brain regions. This is expected to be important for producing experiences of pain in some vertebrates. So we might expect if insects don't have those direct connections like the vertebrates do, we wouldn't expect them to be able to produce the same kinds of experiences. And lastly, many of you have probably seen an insect do something unusual in response to some kind of injury that it has suffered. So walking on a crushed limb, mantids continuing to be sexually cannibalized, that kind of thing. So this is behavioral evidence that Bastin also relies on in his publication. Insects don't seem to respond to mechanical injuries in ways we might expect, in ways similar to vertebrates that are clearly experiencing pain. So this paper is from 1984. It is 2023. That is 40 years of science that has gone by. We have learned a lot about insect neurobiology and behavior in the past 40 years, yet we tend to rely still on this Eisman paper as our definitive answer on whether or not insects can feel pain. This is problematic because almost everything in it is wrong. One problem, nociceptors exist in insects. We have found them, they have homologous nociceptive ion channels to the vertebrates. Direct connections have been found between those specific sensory integrated brain regions that Shelley Adamo pointed out in 2019 as central to her argument, the central complex and the mushroom bodies. In 2020, we did the first connectome of the adult Drosophila fruit fly, found direct connections. So that hypothesis lasted about 6 months. In terms of the behavioral evidence, walking on crushed limbs, I think that actually is probably the most compelling piece in the whole bit, in that like it is true that we can see insects do those things for sure, but there are many reasons why we might choose not to actually place much faith in that evidence. One would be we know many sentient beings like vertebrates, I think about prey animals like cats, they can be wounded and behave as though they are not wounded. So there are other motivations in play for animals that might change how they behaviorally respond to injury. Another thing is that insects have exoskeletons. Exoskeletons are quite different from endoskeletons. You might expect that that could affect their protective responses to the kinds of injuries that they receive that have to do with mechanical stresses and force. The last thing we might consider is that there are many kinds of pain. I told you earlier about the pain that you feel when you touch a hot stove. We all know that insects respond significantly to heat stress and to shocks. So we do see them not behave normally in response to some kinds of pains. There's no reason to privilege mechanical stress injuries over heat-based damage or other kinds of injuries that insects might receive per se when thinking about whether they can feel pain in response to any stimuli at all. So this is not to say that I'm like, well, Eisman was wrong, insects are sentient, it's all over. Instead, I'm saying we should probably not rely on that paper, and we should instead go looking for what we think are, a priori, critically important neurobiological features and behavioral lines of evidence. And then once we kind of have a framework for assessing whether we think animals are likely to be sentient or not, then we apply that framework to insects and see where they come out. So the Birch group at London School of Economics went through and did this for an invertebrate sentience framework. It has 4 neurobiological criteria, 4 behavioral criteria. I do not have time in the 25 minutes that I have today to go through all of it, but I recommend that you read this publication to understand why they chose these particular criteria. And I'll have you know that they are kind of like long-standing criteria that have been used for assessing vertebrate sentience as well. They rely very heavily on the Smith and Boyd 1991 framework to develop this one for invertebrates. They then used this framework in the initial publication to assess the evidence for a more accepted class of invertebrates that most people tend to think are highly likely to be capable of feeling pain, which is the cephalopods. So how do they come out when we look at this framework? Well, they meet 7 of 8 criteria to a high or very high level of confidence, which this framework says provides very strong evidence for sentience. Decapod crustaceans, so that's like your crabs and your lobsters, they come out at 5 of 8 to a high or very high degree of confidence, which this framework rates as strong evidence for sentience. And what's important to note here is that this is an action-relevant framework. So the UK government in 2022 chose to include both of these groups in their animal sentience bill because of the evidence that was accumulated in this particular framework setting. So with Andrew Crank, who is here today, the Birch Lab group, and led by Matilda Gibbons, who's a phenomenal postdoctoral scholar now in the States, we went through and we applied this particular framework to adult insects and juvenile insects across 6 different insect orders. Only gonna have time today to talk about the adults, but if you're interested in this work, there's a 75-page paper where we review over 350 studies that you can read in Advances in Insect Physiology, which is summed up basically by this table. So what we find, I'll call attention to a few key details. In the Latodia and the Diptera, so that's your cockroaches and termites and your flies and mosquitoes, there are common model species, the American cockroach and the fruit fly. In those 2, because they're particularly well studied, we find that they meet 6 of 8 criteria to a high or very high degree of confidence. So that is more than the decapod crustaceans, but 1 less than the cephalopods. Still quite close. For the 2 criteria that they don't meet to a high or very high degree of confidence, you'll see that the bars are saying very low, no research found. So that means not a single study was found that could guide us either way in understanding whether or not they were likely to fulfill that criterion. So big research gaps that could be filled there. In fact, there was only 1 study that made us not very confident that that insect could potentially fill that particular criteria. So that's for Hymenoptera for criterion 8, which is analgesia preference. So this particular criterion is if the animal gets an injury, does it seem to prefer analgesics in that state? So for this study, they gave honeybees a leg injury and then gave them a morphine feeder and a sucrose-based feeder and said, do they seem to preferentially drink from the morphine-based feeder? And the answer was no. There's a couple of problems with this. Insects don't have opioid receptors, so morphine is not a validated analgesic for insects. So that makes it hard to feel that this is actually confidently giving us any evidence at all about analgesia preference, but we included it for completeness. I'll also note that insects do find morphine aversive from a taste perspective. So now you're asking them, do you prefer something that may not be a validated analgesic for you, that also makes you taste bitter things over sugar water when you may or may not be feeling pain? I think it's a poor choice of experimental paradigm. It doesn't give us much information, but we included it for completeness sake. Probably one of the most important results that is sort of hidden in this table is how much gray there is. And the gray, right, is representing very low no research in most cases. So what that means is there are huge evidentiary gaps here that are going to lead us to necessarily feel quite uncertain for the vast majority of insects, whether or not we think they are likely to be sentient and have the capacity for pain. But this is a morally urgent question. So what do we do when we have significant empirical uncertainty and yet moral urgency for how we act? Well, we ask ethicists. This is where the interdisciplinarity of this topic comes in so well. So ethicists, what do we do when we have uncertainty? Many times they will recommend we apply a version of the precautionary principle. This is the idea that if you are uncertain, if you may be causing moral harm, you should act as though you are causing moral harm. Now, there are strong versions that you can make of precautionary principles and weak versions. Strong versions are things like you treat the insects as though they definitely are sentient until you know otherwise. This would lead to things like regulated harm minimization protocols. But you can formulate much weaker versions of precautionary principles as well, where you treat insects as though they may feel pain while you're uncertain, and you even proportion the level of your caution to the level of evidence that you may have. So you might imagine this leading to personal harm reduction approaches taken in research and welfare improvements in farming by individual producers. And it's this second weaker version that I think most folks have actually been recommending in the entomological community, because there have been many calls over the years for precautionary action in insect research, including by some folks in this room here today. But I think one of the most interesting things to me is actually Eisman et al. In 1984 call for precautionary approaches to the treatment of insects in research. So they recognize at the end of their paper that although they don't think the evidence they have available to them at that time suggests that there's an adaptive role for pain perception in insects, there is still a possibility that that could be true based on the evidence. And so they recommend that insects have their nervous systems inactivated prior to traumatizing manipulation. That is an example of the precautionary principle in action. So one thing I think has been a challenge for our field, the reason why we haven't taken that precautionary call to action as seriously as maybe we should have over the last 4 decades, is because nobody really knows what to do or how to do it. How do we actually improve things for the welfare of insects in a precautionary way? So this is something I tried to address this year with the Insect Welfare Research Society. We gathered an interdisciplinary team of welfare scientists, entomologists, and ethicists, and we went through and using guidelines for protecting and promoting welfare in vertebrate species, we went through and we basically developed our own guidelines for protecting and promoting insect welfare and research, which you can find on our website. We'll be adapting them every single year so that they kind of meet with the best science on insect welfare. And they're just a general framework, so they're necessarily quite vague. That's because the actual specifics of how you promote a specific species welfare is going to vary from species to species. But the general principles, the framework of the things you might think about, enrichment opportunities, nutrition, housing, methods of euthanasia, humane endpoints, those kinds of things are not going to vary from species to species. Those considerations can be taken into account across the insect welfare research space. So now I'm going to transition into a specific use case where insects are used in farming as food and feed. For those of you that aren't familiar with this industry, it's a rapidly growing industry because insects are a protein source. They can be eaten by humans. There are 2,000 species known to be edible to humans, and they can also be consumed by a variety of farm vertebrate animals. And the reason this industry is of such high interest right now is because we have a growing human population with ever-increasing protein demands. We're running out of sustainable ways to farm animals to feed that growing population. It's becoming quite urgent, and so we need more sustainable sources of protein than the farm vertebrates we've relied on in the past. Insects may be able to fit that bill. The last thing I want to note is that this is a global industry, so there's a lot of variation in the scale of facilities and the practices that are used and the degree of automation. We are not going to have time to talk about all of that today, so I'm really going to be very brief in my review of this topic. So when we think about the welfare of individuals, we need to know how many individuals are actually being impacted by a particular practice. We need to estimate the scale of the industry. So the most recent estimate of the industry scale is from 2020. That means it's probably a vast underestimate because, as you'll see in a moment, the industry is growing quite rapidly. But it puts the number of farmed insects in this industry at about 1 trillion to 1.2 trillion a year across the globe. To put that into perspective, this is terrestrial vertebrate farming numbers. So cows, pigs, chickens, goats, sheep, etc. all fall into this little green bar right here. All of factory farming for vertebrates is this green bar. And then these are the insects as of 2020. So we are talking an explosion in the number of individual animals that are being farmed in massive, intensive production facilities as a result of this particular industry. And it is growing extremely rapidly. This graph is the new investment capital flowing to the industry. In 2015, we're looking at about $20 million US. By 2020, we're looking at over $420 million US of investment capital. This is because there are expected to be very new markets that are going to increase demand for this product substantially, in particular fish meal replacements, where currently we feed farmed fish with fish, but we could feed them with insect protein instead. The UN has estimated we could safely replace 100% of fish meal with insect protein. But if we take a more conservative number first, just 25%, that would result in 40 to 80 trillion additional insect deaths per year in this industry, which looks something like that lower red bar on this graph, where that orange arrow is pointing to the 2020 numbers. If we replaced 100%, it would be the larger of those two red bars. And the important thing to note here is that those two red bars are just considering fish meal replacements. They're not considering poultry feed, they're not considering pig feed, they're not considering pet food, and they're not considering human consumption. All of those other markets would need to be added on top of those red bars. So we are talking about truly an explosion in the number of individual animals that are being used in this industry. So when we think about the scale of the industry and we think about the kind of level of evidence that we have for the possible sentience of the beings that are being farmed, it becomes maybe imperative to start thinking about how we could improve their lives under these conditions. So we want to investigate their welfare and try to improve practices that will improve their welfare in farms. So this is the research area that I spent most of my time thinking about. This publication of mine came out last year in Journal of Insects, Food and Feed with Shafanya Chia, Bob Fisher, and Jeffrey Tomberlin. And we basically used black soldier flies as a case study to understand welfare concerns in insect farming. So we go through and we look at all of these variables that you see here listed on the screen that could affect welfare in farmed black soldier flies under the conditions in which they're normally reared. Now, most of you in this room are probably aware, not a lot of research out there specifically on insect welfare, so you might think this review would be very short. You would be wrong, it is like 40 pages. And that is because there is one indicator of welfare that does get studied quite a lot, which is population level mortality, where you can assume that if like 70% of the population has died under some particular condition, probably not a high welfare condition. So that is the metric that we use for most of this paper. It's just like, how is the population faring on the level of survival? And even that gave us many current welfare concerns, future welfare concerns that you can read about in the publication. These are the 8 current major welfare concerns we identify. I'm going to very briefly discuss just this one of them with my remaining time, which is non-instantaneous slaughter without stunning or anesthesia, which is currently practiced in the industry at scale. I'm focusing on this one because I think it's one the public is actually most interested in. Also, a lot of producers themselves have demonstrated interest in this topic, and it's one that I am trying to collect empirical research on to demonstrate how we can help producers improve their practices from a welfare focus. So there are many methods of slaughtering black soldier fly larvae. These include things like grinding and shredding, boiling, baking in ovens, microwaves, asphyxiation, and others. You can imagine just from that list, some of them are more instantaneous, some of them are much less instantaneous. It takes many minutes to microwave an insect. It can take several hours to bake them in a convection oven, depending on the conditions. Some of them might be more likely to be instantaneous, like, for example, grinding and shredding, or potentially boiling. But that's going to depend on your standard operating procedures and the way that you implement those techniques in farms. So I have conducted a research project in progress right now with some folks at Texas A&M University, where we're looking at kind of identifying what variables in standard operating procedures and equipment design could actually influence the humaneness of particular slaughter methods at scale. So we are looking particularly at grinding right now. This is a classic meat grinder. You can see this is the particle size plate that kind of is at the end of it. So basically the larvae get forced through that particle size plate, and that's what's going to cause the mechanical disruption of their body into particular size pieces. Now, as we have discussed, insects can actually sustain significant bodily injury and come out of it still alive. So sometimes they come out of this machine still alive, but in pieces, and sometimes they come out completely and totally dead. We would like them to come out completely and totally dead. That would be the instantaneous and more humane condition. So we want to know what variables might affect that. One thing that might affect it is the size of these holes in the particle size plate, where you might imagine that bigger ones obviously allow larvae to come through, more likely to be in larger pieces that are still alive. So we have tested this. We've gone through and looked at large, medium, and small diameter particle size plate holes from 12 millimeters down to 2.55. And there are multiple possible ways larvae can experience this, but I'm just going to sum it up in some specific numbers here. So the likelihood of humane death in the large condition is 62%. It goes up to about 82% in the small condition. And we've actually also uncovered in kind of our secondary trials, because this was the pilot data from our first experiments last fall, some other things that we can do that increase the likelihood of humane death up to about 99%. So these are the kinds of empirical recommendations we can make to producers to say, look, you can increase the likelihood of humane death in your facilities from 62% to 99%. And that is a substantial improvement for the animals in question. So I want to end here on kind of like a little bit of a hopeful note, which is that I think we have some really solid foundations for this field of insect welfare and ethics. It's growing, it's emerging, we have some solid foundations. And some of that is actually in all these research gaps that I've been talking about throughout this conversation today. So one example, of course, is that paper I was telling you about insect pain and sentience. All of those gray boxes, it's really good we've identified those because now we know where we're uncertain, how we're uncertain, what kind of evidence we need to get to increase our certainty for particular orders of insects in particular contexts. Same thing for these welfare studies. I talked about how, we're kind of using this population level mortality metric. Even doing that, we have identified some things that need to be researched. Obviously, those papers can also guide us to understand what other urgent future research directions might exist for insects and farm conditions. And the same thing with these guidelines. If you go and read them and you think about using them in your lab settings, you'll notice that there are some areas throughout the guidelines where we kind of specifically say, we don't really know what recommendation to make for this method of euthanasia at this time because there's not the research there to tell us whether this is a good method for welfare or a bad one. Those are key future research directions on which we can build this field. So there are a lot of opportunities for growth here. A lot of these can be viewed as challenges, things like how do we handle generalizability with the sheer amount of insect diversity that we have in front of us. These are challenges that even though there are a lot of fish species out there, got nothing on insect diversity, right? These are challenges for our field, but there are also opportunities for us to understand these organisms from a very different perspective. And I'll say that I'll echo kind of calls by Eleanor, also by Emily earlier today about coordinating across disciplines and contexts being so fundamental to this field specifically. That is something that I hope the Insect Welfare Research Society will help with. So if you visit us at insectwelfare.com, you'll find that our board is already ethicists, welfare scientists, and entomologists. We plan to keep it that way. We hope to serve all of those communities because this is something where we need all of those disciplines informing our approach to improving insect lives and using them ethically in a variety of contexts. We also need the people who are practitioners of insect use to be involved in this conversation. So in the research context, that's really easy because the entomologists are doing the research and the practitioners. But think about the farming context. Producers need to be in the conversation with us, right? Because they're the ones who actually know what the practices on farms are, what the welfare impacts for those animals are likely to be. So we need to be building those bridges to connect across contexts and across disciplines. And lastly, I'll just plug this, that I talked a lot today about my interests in ethical insect use from the welfare perspective, but there are actually many other ethical issues that manifest in entomology that we often don't get to discuss enough. From things as mundane as like publication ethics to the ethics of de-extinction research to the ethics of gene drives and release of GMO mosquitoes, all of those, right, are going to be ethical issues that we face. So the American Entomologist, which is a publication by the Entomological Society of America, has recently allowed Dr. Bob Fisher, who is an ethicist at Texas State University, and myself to begin this column called Ethos, where every single issue we tackle some entomological challenge. Some of them might be related to welfare. Our next one is publication ethics, very mundane. I hope you love it if you choose to read it. But you can also use our Ask an Ethicist feature where you email us at that email address. And like any help column, we will try to answer those questions in future columns. Moving forward, next year, we also hope to have other collaborative pairs of entomologists and ethicists writing for the column moving forward. So with that, I want to thank you for your time and attention. You can get in touch with me, obviously here today, also using this email, my Twitter account. You can find me at the Insect Welfare Research Society, and I would love to take any questions. Any questions? I've got one to start. Oh, yes, go for it.
How do you know when an insect is dead?
That's a great question. So, and a really challenging one. So thanks for starting us out with a really hard thing. So we are using as our metric of mortality the loss of motor function, of non-stereotyped motor function. So we do not have the capacity to be doing like actual nervous system testing. Like you could do electrophysiology, for example, and say like, is the nervous system still operational or not in the brain proper? We do not have the capacity to do that in this particular experiment, but often for animals, what you use is a measure of like behavioral loss. So for this experiment, basically imagine that you cut the insect in half. Is the body still engaged in motion that is goal directed, or is it just like if you poke it, it like reflexively jumps? The other thing you can look for is whether or not the brain proper is still in the body segment. So we know where the brain is located. The ventral nerve cord can be separated from the brain tissue itself. If the ventral nerve cord is solely responsible for responding, you often get like reflexive jump-like responses that we would consider to be basically, like death to the point of caring about the subjective experience of pain. If the brain is still in there and you're seeing those goal-directed motions, then I think you have more confidence the animal is not dead.
Yeah, but many insects, if you chop their heads off, still carry on as normal.
Carry on as normal.
Well, I mean, for praying mantis, the head normally comes off the male first. and yet it still mates.
So it still mates. This is a great point. It still mates.
Is
but
it
does
dead
not-
or not?
Well, I would say it is not dead in that the brain, which is the part that we care about for the subjective experience of pain, that is dead if the head has been eaten. So then, yes, that's a dead animal. I mean, like a headless chicken would be considered
Yeah.
running around dead to the point of subjective experience. Is the nervous system that is run by the spinal or ventral nerve cord still functional? Yes, but we don't consider that relevant to the subjective experience of pain.
Right, okay.
We're assuming the processing power is centralized in the insect.
We are, well, so what do you mean when you say centralized?
Well, brain tissue, nervous tissue in the insect goes down the entire body. We're assuming the processing power is in, to actually perceive pain is in the head.
We are assuming that, and in general, that's because that kind of assumption is broadly true across many other organisms that also have their nervous system distributed throughout their body. So it is possible that the abdominal ganglia, for example, could have some kind of sensory integration functionality. We discussed that a little bit in the paper, but when you look at insects and the central processing sensory integration regions in the brain, you get connections that are much more similar to central processing in vertebrates. And you get a lot more sensory integration happening in those regions. We also know there's ascending and descending control of locomotion that happens in the brain proper that is controlling goal-directed kinds of behavior as compared to reflexive responses. Again, very similar to what happens in vertebrates.
I do think it's an assumption, though. I'm not sure I agree, that, well, because you can see it in centipedes, for instance. I know not an insect, but if so when I conduct euthanasia in a centipede, I give a series of injections down the entire length of the body because you can see that a bit down, further down the body will still respond to stimuli. Now, whether that means it's just a reflexive response, whether it's a processing response, I'm just not sure that the assumption is a good one.
Also, I think, let me give you an example. I don't know much about centipedes. I will give you an example from cockroaches. So in cockroaches, we know that they have different responses to, like different escape behavior responses based on whether their head is attached or not attached. Just like you might find in like decerebrate mammals or in chickens with their head cut off, they are capable of creating motion and having some reflexive responses to stimuli, even with the head entirely removed in all of these groups of animals. But when you put the head back on, you get very different kinds of responses, right? So cockroaches, their startle reflexive response is non-complete without the head attached because they lack goal-directed centralized locomotory control. When you have the head attached, you get very different goal-directed escape responses. So we know they are capable of motor function without the head attached, but it is not the same kinds of responses. It's much more reflexive, much less goal-directed. And I think that plays into, across all of these animals, the likelihood of centralization.
I think it's a very good point. I'm just not sure you could assume there's no goal orientated in the rest of the body. I mean, there's a very interesting video of a wasp with its head removed, and then it picks up its own head and flies off with it. So I just think it's probably something we need to ask more questions about. But I can see that actually there's a lot you don't know, and I certainly don't myself.
Well, I would agree that for you as well, that I am not attempting to say that I am definitively certain that there is no additional processing in other ganglia, just that I think the processing that happens in the head ganglia is especially important.
This sounds like an excellent conversation to take forward as well. Does anybody else have any specific questions? Yes.
I'm just wondering, very, very good talk, by the way. Have you limited yourself to insects because others started to develop crustaceans and other groups? Or is it because you're not interested in them, or is there another reason?
Yes, I mean, anything that gets wet, I'm just completely out. So that is actually a great point. So we are called the Insect Welfare Research Society, but we also are interested in other understudied invertebrates, which I tend to take as like literally anything that isn't a cephalopod at this time. So sorry to be a cephalopod hater for everybody who loves my octopus teacher, but unfortunately they get a lot of attention in this way, and other groups like shrimp, like decapod crustaceans more broadly, centipedes, millipedes, your myriapods, your chelicerids, all of those don't get attention, and we are open to covering all of them. We framed it this way because I thought Invertebrate Welfare Research Society was actually much too broad. Arthropod starts to like confuse people, like what is an arthropod? I feel like I've never heard that word before. So I was like, we'll call it insect, but we will hone all of the researchers interested in all of those arthropod groups.
I've got a few questions from our viewers online as well. So the first one is from Fran. Do you know of any research into handling pet insects and their welfare?
That's a good question. So I don't know any research into that question specifically. I actually imagine that to some extent the research guidelines will be useful to you in terms of like rearing and things like that. Obviously, probably a little bit less about experimental design is relevant to that particular context. But as far as I know, nothing on them as companion animals. Yeah.
So another good research, yeah. Lee Haynes says, again, a lot of questions through. This is great. Lee Haynes says, how willing are insect farmers to engage in insect ethics discussions?
I would say, I know people have this sense that animal agricultural producers are very unfriendly to conversations about welfare. That has not been the case for me discussing this topic with producers. I think they understand that this is a conversation that like they need to be having. They're a little uncertain about how to do it. They're looking for empirical guidance. The International Platform for the Insects is Food and Feed, which is Europe's kind of based group, they actually have already issued in 2019 their own report on welfare calling for additional research and guidelines. NICEA, which is the North American one, is interested in having that conversation. The NSF Center for Environmental Sustainability through Insect Farming, which like helps all of the producers globally, invited me to give a talk on insect welfare that the producers were extremely friendly towards me and very excited about the research topic. I think one thing that helps there is that there are many reasons that welfare science can benefit producers, especially in an emerging industry. You know, I told you mortality is our metric. Mortality for producers is economic productivity loss. So we're actually kind of speaking the same language, just from different perspectives right now, where I'm concerned about the individual animal, and I hope to help improve its chances of survival. And they're like, I also hope to improve its chances of survival. That would be great. So I think there's like a feeling of kind of communion right now, at least, that insect welfare is a way to help them establish a robust and also ethical industry. And I'll make one last note on that, which is that this is an industry that really relies on being ethical. It sells itself as being sustainable, better for the world. And I think they care about the stewardship of resources. And whether you see the insects as a resource or as an individual, you care about stewardship from that perspective.
Great. So we've got one from, I'll take one more from online and then one more from the room, because I don't want to hold the limelight. So we have one from Jonathan Amory. In farms, livestock welfare improvements have been easier to drive when linked to better production. Are there any potential candidates for insects considering the high mortality rate? Which kind of you pretty much answered in your piece.
Yeah.
Anything else you want to add on to that?
There are some specifics that I've been talking with producers about. So one example would be that in black soldier flies, there's this misconception that the adults don't eat. And so right now they're starved to death in industry conditions. It turns out if you feed them, you get dramatic increases in reproductive output. And also like more genetic mixing and things like that, which can be good to prevent inbreeding depression that happens on farms at these scales. So figuring out what they eat and how beneficial those different kinds of like protein and carbohydrate compositions actually improve reproductive output is one thing that I would say is like a pretty obvious thing to be able to do for producers. Another thing would be stocking densities for mealworms often cause high mortality. Figuring out what the appropriate stocking densities are and how you can change other factors like crate design to improve survival under those conditions. Another obvious one. One that I thought was really cool when I talked with producers that they were super interested in is this phenomenon called shipping sickness in crickets, where if you ever order crickets to feed to a pet reptile, you'll get like 50% of them dead when the container comes in. They call that shipping sickness. They have no idea what causes it. It's just like 50% mortality. You have to send double the crickets the person orders. Yes, that is right. So crickets use vibrational cues to assess the likelihood of predators being around, and it's been demonstrated that increases octopamine levels. If you like vibrate them, it increases their octopamine stress levels in their brains. You can imagine that when you are being transported on a truck, the entire time is this experience. That would be extremely stressful for you if this usually means I'm about to get eaten. So that might account for why we're seeing so much shipping sickness. It could change the way we do shipping product design, for example. So I think these are all avenues for investigation that once you see the perspective of the individual animal as an individual, you might be able to make improvements that would increase economic productivity.
Great. Another question from the room? No? Okay, well, in that case, we've got two more questions, and then I think we're running a bit ahead of time, so we can have a little break, grab a cup of tea, and then get back to the online sessions. That's like good? Cool. Okay, so your last two set questions. We have one from David Fisher. Insects are phenomenally diverse, which creates big problems for coming up with guidelines. Do you think that we can apply recommendations across higher taxonomic levels, as then we might for vertebrates? I.e., is there good evidence that one beetle species feels pain? Can we apply that to all other species or other insects?
This is a great, very challenging question about generalizability in insects. So I'm going to tackle it differently for sentience versus for welfare. On the welfare side, so like the actual recommendations you make for a specific species, generalizing is somewhat dangerous. And that's just because different species have very different life histories and ecologies. Even sometimes across life stages within a species, you get very different life history strategy or like ecologies. The things they eat differ, stuff like that, where they live, the conditions they thrive under. And so you need to be really careful to consider the specific species in that case to make sure that you're actually provisioning for its specific welfare. For example, there are insect species, moths, that don't eat at all as adult silkworm moths, classically. It would be bad to apply a recommendation to not feed your adult silkworm moths to all other lepidopterans that eat at the adult life stage. So we should be very careful to be species specific there, or at least some lower taxonomic level than order there. When it comes to the generalizability on the sentient side, I think the question gets much more complicated. I will say that the neurobiological evidence is actually, a lot of it's quite ancestral. So for example, criterion 2 is sensory integration regions, higher order sensory integration regions. We know that going all the way back to the Archaeognathia, that you get mushroom bodies, which are a higher order sensory integration region. So we can be pretty confident that we can generalize on that one at adult life stages across insects. Even ones that maybe haven't been as well studied, we can probably feel pretty confident about doing that. But for the behavioral criteria, I think that's a little bit harder to say, given the data we have at this time, because the evidence that we have is very spotty across just a couple of taxa. And there are reasons to suspect that behavior might differ strongly based on things like life history strategy. I think we run the risk of falling into two possible traps. One is we focus so much on individual species that we refuse to generalize, and then at all. And so then we don't protect animals that should be protected on the basis of that evidence because of the uncertainty about how to generalize. The other side of it is that we get so interested in just trying to figure out everybody all at once, like Iceland did, that we really lose out on key specific information that is relevant for distinguishing different species from one another and their capacity and likelihood for sentience. So I think somewhere we're going to need to figure out how to manage that uncertainty. But I think that's harder to figure out on the sentience side than the welfare side.
And final question here. So we have one from Richard H. Natural biocontrol might comprise of a parasitoid injecting an egg and then the larva eating its prey slowly from the inside. This is completely natural, but sounds horrible. Should we be discouraging biocontrol? That's a good question.
This is a good question. So there's multiple things here. I mean, discouraging. So first of all, something being natural doesn't mean it's high welfare. I just want to really clearly state that up front. We should distinguish between those two notions. Acts of predation, acts of parasitism are very natural, and nature is not a high welfare place. So from a welfare perspective then, we wouldn't be jazzed about things that are natural but that are bad for welfare. That said, there are many reasons, these questions, the ethics of it, are complicated trade-offs. So as one example of that, imagine, there are lots of insects that are, pests of crops. We could kill them with pesticides and then eat the crops, or we could not kill them with pesticides and then all starve to death. There's a trade-off there between our welfare and insect welfare. I think we all know whose welfare we're going to prioritize on this particular trade-off. We do this all the time though, right? We do this with mice that are rodent pests in fields and rodent pests of crops. We do this all the time, that we make these trade-offs where we prioritize particular economic goals, knowledge production goals, welfare of human goals over the goals of other, or the welfare needs of other species. So we should not be afraid to have the conversation, because it doesn't have to lead us to everybody starves to death and nature just runs away with it. As far as the parasitoid thing, I think some considerations I would give it there then is do we have other methods of control that are higher welfare? Well, then maybe we should implement those first instead of rearing large numbers of insects to then inject other insects with parasitoids. The other thing is that often parasitoids actually have mechanisms for controlling the behavior of the species that they are parasitizing. So it is not always the case that just because you have a disease or a parasite, you actually have lower welfare as a result of having that disease or parasite from your own subjective perspective. There's a fungus that takes over the butts of cicadas and eats them, but they don't seem to actually mind it, and it has some effect on their sexual behaviors and controls. We know that's common in parasites. So I think that's also one thing to consider, is I don't have enough information about this specific parasitoid situation to know whether or not it's actually affecting the subjective experience of, say, the lepidopteran pest that is being consumed. It feels like it should be bad, of course, to us intuitively, but just because it feels like it should be bad intuitively doesn't mean we actually know that it is. It should probably be tested. But yeah, if we have higher welfare and more instantaneous methods of killing those pests, maybe we should prioritize those and that line of research.
Great. Any more questions in the room? No? Oh, yes, Ollie.
I've got one, yeah. So you mentioned about publication ethics before, as if it was boring. So given that this is the last question, I just wanted to ask you a little bit more about that and what you think publishers and journals should be doing to support this kind of work that you have with these guidelines. Perhaps we should be referring people to these guidelines in terms of publishing. Perhaps we should be having statements and papers around this.
Great question. This is great. I didn't plant this question. I will say the publication ethics paper that is coming out is not about this specific question. It's about like who should be an author on a publication,
Okay,
how we
cool.
decide on authorship decisions.
Also cool. But
Also cool, also very cool, yes. But this is still a great question. So we're actually hoping to get some feedback from the community of the Entomological Society of America on this at the upcoming ESA meeting.
Okay,
We're
great.
going to ask people what they think about, like, would they be interested in the society taking a more active role
Yeah.
in kind of guiding folks to like visiting these guidelines and stuff like that. My personal perspective on it right now is that, like, yes, I do think it would be ideal from multiple instrumental as well as intrinsic value of the insect's reasons for societies to take a pretty strong role in guiding the conversation on welfare. I think this because I expect we have somewhere between 2 and 5 years before there is a lot of interest in insect welfare from the public, from legislators. It's growing for the farming industry, it's coming for research settings.
Yeah.
The public is growing, like you mentioned, in their care for insects and seeing them as individuals with value. We do not want to be caught on the back foot in that conversation where they're like, look at these atrocious researchers that never care about insects and are doing horrible, terrible things.
Yeah.
That is not for us, the way our community wants to be put on the back foot, being reactionary in that conversation instead of being proactive. Much better is if we can go to the public and we can say, we also care about this issue. We have guidelines, we require ethics statements in our journals, etc. Animal Behavior Society has already taken this approach. If you go to publish in their journals, even as an entomologist, you have to include an ethics statement. I'm working to develop tools that I think journals could use or societies could use to guide that conversation. So like one of those is a paper that I'm hoping will come out later this year, where we basically go through old entomology papers and pull out where people are actually reporting on ethics and just not calling it ethics. Like people often report that they anesthetize their insects with cold prior to doing traumatic manipulations. That's welfare improvement that you're just doing as part of your standard practice. You could report on that as a welfare improvement. It would be good to do that from an ethics perspective. And then yes, I think like guiding people towards these guidelines, asking for optional inclusion of ethics statements in journal articles that are published, all of that could be a way that journals and societies could steer the community towards engaging in higher welfare practice, and that would benefit the community in the long run as this conversation develops.
Yeah, fantastic. Completely agree, and I think something that we could work with the ESA on as that area. So yeah, it'd be great to chat more.
That would be great. I love it.
Yeah.
Great. All righty. Thanks so much, everybody.
Thank you so much for answering so many questions.
Oh, it's lovely.
There's a few more questions, but I feel like everybody
I'm ready.
needs a break.
Oh,
Needs a break.
okay.
Would you be happy for people to email you questions
Of course.
or things like that?
Yeah,
Okay.
Inbox is always open, director@insectwelfare.com. There's like 50 emails you can find for me online. Just pick any one of them.
Brilliant. So anybody who's still got some questions online, do feel free to email Megan. All right, does everybody want to have a quick 5-minute break, go grab a cup of tea, and thank you again.
Thanks so
And then we'll
much.
be back for the next bunch of talks. Brilliant. Thank you. Great. Oh, thank you. That gives us a chance to get the online. The next section. It still scares me. But so the next section is going to be a series of flash talks. Each speaker has 5 minutes. So we won't be taking any questions after each one of the flash talks. But if there's time at the end of this particular session, we can have a few questions there. So if you have burning questions, then make a note of them, and then we'll have them at the end. Also, just something else to note, in this particular section, we have a student award. So we have 2 undergraduate speakers who are speaking today, and we have a brilliant John Avery who's online and has very, very kindly offered to judge them. So we will be announcing the student presentation at the end. No pressure, no pressure. So we've got our first speaker, which is David Fisher. Are you online, David?
Yes, I am. You can hear me.
Excellent. Okay, so I think if you just share your screen, the floor is yours.
Yeah, and I've got 5 minutes rather than 3. Is that right?
Yes, you have a total of 5 minutes. You do.
Exciting. All right. Hi. So my name is David. I'm at the University of Aberdeen. So this work is on motivational trade-offs in a cockroach. So I'm very excited to be able to fill in one of those gaps that was highlighted in the previous talk. So this work was conducted by Joe Jenkinson as part of his honours project. So we're interested in the moment in whether invertebrates feel pain, and this is in part because we see organisms such as octopuses engaging in really cognitively sophisticated behaviours, which makes us wonder about other aspects of their lives. And it's also because we're increasingly starting to farm invertebrates, and so aspects of their welfare come under focus. So one of the things you might want to be interested in looking at when determining whether a stimulus is painful to an organism is whether the organism engages in motivational trade-offs in order to avoid that stimulus, and so sacrifice obtaining something they might otherwise desire in order to avoid being exposed to that stimulus. So light is a really excellent potential stimulus to use, because we know organisms such as cockroaches are keen to avoid it, and so we can study motivational trade-offs using light as a potential noxious stimulus. And while researching this, I came across this study from 1912 by Zmansky, and it's interesting to hear Ellen talk about looking at old research. So this paper, they trained cockroaches to prefer light rather than darkness by giving them electric shocks. I think it's and all 10 cockroaches this person studied learned this. It's a really interesting example of the kind of things that people were doing quite a long time ago. So to do this, Blapticadubia, we have cockroaches in 2 different treatments. So in each treatment, the cockroach had access to a shelter and a piece of carrot as their food item. And then the carrot was either sheltered, so it's underneath a cover here, or the carrot was exposed, so it was out in the open here. And we're going to compare these 2 treatments to see their relative preference for the carrot changes with exposure to light, to see how they might be trading off access to the carrot to avoid exposure to the light. Then we can further probe the conditions that alter this motivational trade-off by decreasing the strength of the noxious stimulus by putting translucent paper over the exposed treatment, or we can increase their motivation to access the carrot by depriving them of food 48 hours rather than 24, which would then increase their willingness to expose themselves to light. So these graphs here show the relative preference for carrot on the y-axis, and so this is the first experiment in the full light treatment, and you can see that in the exposed treatment, when there was light, they're less willing to access the carrot than in the shelter condition, although this difference isn't quite statistically significant. The difference does become statistically significant when you deprive them of food. So if you increase their motivation to access the food, they're more willing to, they increase their willingness to access the food, but not in the exposed condition. So the light still is overriding that willingness. And then if you reduce the degree of light with the translucent paper, you see a much smaller difference between the exposed and the shelter conditions. So there's no significant difference between these 2 treatments, the reduced light or full light, but there is a significant difference if you deprive them of food. And this is true whether I analyze it with a series of Malwin U-tests, which are the results here, or if we fit a GLM and we use an interaction between experiment and treatment. So the conclusion then, we're happy that light is a noxious stimulus for Blapticadubia, and so they will avoid accessing a resource like a carrot if to avoid to stay in the dark and avoid accessing the light. And we can then change the degree of motivation, and that then changes how they make this trade-off, which suggests a degree of central processing between these different demands they have. And so this has implications for their welfare and their husbandry. So we know that exposing them to light is causing them some kind of stress, and so we should minimize that whenever we can. And then we're designing the conditions that keep them husbandry. We can then provide them plenty of shelters and things like that, so that they can avoid light whenever they choose. Thank you very much.
Enthusiasm, thank you very much. Do you want to come up, Casey? Brilliant. Okay. Slides up. Perfect, when you're ready.
Hi everyone, my name is Katie and I'm here to talk to you today about part of my dissertation, whereby which I carried out a study on the durability of different paint types when marking cockroaches, and I undertook that at the Titchmarsh Centre here at Writtle University College. You gonna behave?
We can change the slides.
Sorry, technology. This paint study was carried out as a part of a larger study in my dissertation where I carried out a personality study in cockroaches. And I hit a sort of roadblock when I came to carry out my personality study, as I needed to be able to individually identify cockroaches to know who was carrying out each behaviour. But when researching, I couldn't find any recent or accessible research which described how best to mark cockroaches in terms of durability and mortality rates. And I saw this as a kind of welfare issue because I didn't want to just go put in any old paint or other marking onto the cockroaches in case it caused any health effects or killed them outright. So the main aim of the study was to find a paint type that was the most suitable for marking the cockroaches. So it stayed on really nicely, so we were able to carry out the personality study and be able to identify who was who throughout the study period, and also with the overall aim of increasing welfare in the future by knowing that the paint that we use is not likely to cause mortality or other health concerns. Sorry. So as a part of my study, I used 4 different kinds of paints. I used an oil-based paint pen, which is that one just there. So the application method was a pen, so draw straight onto the pronotum of the cockroach. I used 2 Revell Aquacolor paints. These were both acrylic. One was matte and one was gloss, and they're seen just along the bottom there in the middle. And they were applied using a toothpick and drew the paint directly onto the pronotum of the cockroach again. And the final one was another acrylic paint, the Acrylicos model colour paint, and that was applied using an eyedropper method onto the pronotum of the cockroach. It's important to note that I researched the paints first. I didn't just kind of pluck the paints out of the hat. I researched them, and the oil-based paint pen was used on other species of cockroach, and the other 3 paints were used on wasps and ants in respective studies. We also used a corresponding control group with each paint group, so we had obviously 4 different paint groups for the 4 different types, and we used a corresponding control group with each one because the theory was that if we had a large number of deaths with a paint type that was the cockroaches that were painted, then it was likely the paint that killed them. Whereas if we had a large number of cockroaches dying in control groups, it was expected that it was potentially stress or handling that killed them, or possibly a husbandry issue. So here are the results of my paint study. The paints were all compared to one another using a chi-square test, and so along the y-axis, we've got the paint groups and their corresponding control groups. And along the x-axis, we've got the number of individuals in each category. So the blue colour shows cockroaches that still had a paint spot fully visible on them at the end of the week's observation. The orange colour shows spots that were either partially visible or completely gone from the cockroaches at the end of the week's observation. These 2 categories were merged together to ensure we had the right number in the sample for the chi-square to work properly and be reliable. The grey colour represents control groups. So these cockroaches were never marked as a part of the control group, but they were alive at the end of the week. And yellow represents dead cockroaches. So we had 2 deaths overall in the paint study. We had 1 in the second acrylic group and 1 in the last control group. So there were no major, there was no real significant number of deaths found in the paint study. So overall, the oil-based paint pen was found to be the most durable with a p-value of less than 0.001. We also saw significance in the second acrylic, the Revell Aquacolor in gloss, I think it was, and that was with a p-value of less than 0.05, so that was less significant. The oil-based paint pen was found to be very significant. The oil-based paint pen also had a very low mortality rate. We did have 1 death overall that was not seen in the previous graph. That was found in the personality study. So that paint graph was that was made before we carried out the personality study, so it wasn't relevant to include that in the previous graph. So to conclude with that one, we then used this oil-based paint pen to mark the cockroaches to differentiate them from one another in the personality study. So we have a few opportunities for further study as a result of this study. So it's not known whether or not we could have any sublethal effects of the paint used in the cockroaches. So we don't know if a few days or weeks down the line the cockroaches might have health effects or behavioural changes as a result of being marked with the paint, or they might die a few weeks down the line because of this, but we wouldn't know unless further study was carried out. And further study is also needed to determine the suitability for other species. And it's hoped that this study could inspire future study into looking at welfare issues associated with marking insects and other invertebrates with paint to identify them. And so just a few acknowledgements. Thank you to everyone for listening today. Big thank you to the staff at Titchmarsh Centre and to Dr. John Avery for helping with my statistics, and for my supervisor, Dr. Owen, for everything.
Okay, brilliant. Well, thank you very much for that. Thank you very much. So our next speaker should be Jonathan Edwards. Are you online, John? Do we have Jonathan Edwards? We don't think he's online. Okay. No. Okay. Well, in that case, if Jonathan is not available, then we go straight on to the next talk. Benjamin, are you free to? Brilliant. Okay.
Also, do I have to click it?
Click the thing. Yes, it's been a bit more. Good luck.
Okay, so hello, I'm just going to introduce some preliminary research on thermal nociception in large photophore spiders using the Hargreaves test apparatus. I should be quite clear, I would regard myself as quite a small part of this study. I'm more involved sort of within the analgesic side. The majority of the work has been done by Alice and the trims, as well as everyone else in here. So we'll just get straight into it because time is limited. It's going to work. It's not working. So essentially, our understanding of pain and nociception in invertebrates is poor. I think there's still a lot of studies we need. And really, a lot of the studies that are present are based on small mammalians or mammalian species. Well, the issue we have with invertebrates generally is they're very small, and a lot of those tests that we would use in invertebrates don't always work or translate to invertebrates. So the thinking is perhaps a large ferrophosid spider, which is amongst the larger invertebrates, could be used as a model for pain and nociception in invertebrates. So what we've used is we've repurposed something called a Hargreaves test, which is essentially a heated plate and responds to a heated plate from a mammalian model in mice and rats, and we're trying to apply it to tarantulas, basically with the mind that we then have a model that we can use to regard analgesics, etc., so on and so forth. Next slide. So what this initial bit of research establishes is, what is the baseline response, if there is one, and how conserved is it across species? Now, we don't necessarily have the sample numbers, and this has been done on a research collection of ferrophosid spiders used in venom collection and venom assays. Now, we don't necessarily have sample numbers in ideas of from the perspective of a single species, but we do have sample numbers from the perspective of a number of species, so it's a slightly different approach. So we really wanted to do is just confirm that this is something that could be done, and we wanted to also confirm if it changes with injury. So next slide. Fabulous. And this is basically the setup we've used with different species here. Airflow was stopped. We've got the standard Hargreaves apparatus, left to acclimatise for 5 to 10 minutes in one space, and then we essentially positioned the Hargreaves apparatus below one of the feet, and then tried to essentially took video to get an idea of the speed and of that response. So the reason this is all being done in essentially a fume cupboard is because obviously they have urticating hairs, which can be quite frustrating. Next slide. So this is a video of it being done, just to give you an idea of what we're looking at. So pretty cool, I think. Next slide. And this is the results. So it's pretty conserved across multiple different species. we sort of did get a consistent withdrawal temperature. We go to the next slide again. And we also did a pinprick. And actually we found essentially we had a quicker withdrawal when we had a pinprick on that limb. So next slide again, because I'm being rapid. We've done a very preliminary analgesic study. So we picked something called meloxicam. Meloxicam is a non-steroidal anti-inflammatory drug. It was picked deliberately because I know that vets have anecdotally used it on pet invertebrates in the practical world. And I had a suspicion it probably wouldn't work because spiders and insects don't have COX-1, COX-2. COX-1, COX-2 are a part of the prostaglandin pathway and part of the anti-inflammatory response in mammals. Now, they do have some analogous pathways to that if you do a genomic study on their genomes. However, I wanted to really confirm it did nothing. So this is why we picked this one, and I know it seems a bit odd. I did get ethics approval for this, full ethics approval, because I get ethics approval for everything I do, through the RCVS Ethics Board. So the Royal College of Veterinary Surgeons is the accreditors for veterinarians in this country. There was no difference between negative control and meloxicam dose, and I tested meloxicam dose in locusts to 20 milligrams per kilo, which in translation is about 20 times what we would give in a cat or dog. It's 100 times what we would give in a cat or dog. And in tarantulas up to 2 milligrams per kilo. So very preliminary study, very small sample numbers. So this was really a preliminary study. Next slide. So there's still lots of questions we need to ask. I feel like we have proved that ferrophosid spiders are suitable for use in the Hargreaves thermal nociception assay. I think this is currently, and I would say pre-publication, we need to collect more data, and I think we want to establish an analgesic question. So our next analgesics we're probably going to test will be some of the opioids and some of the local analgesics, particularly procaine or lidocaine. And the other interesting finding is the Grammostola spider, which had the pinprick, continued to have an increased withdrawal threshold for 52 days afterwards. So that continued to be felt for quite a considerable time afterwards. So very brief. Thank you for your attention.
Thank you very much for a great talk. So we have a few minutes. We have a few minutes free. So if anybody has a question for any one of our speakers, has anybody got any? Yes.
I was just wondering about the temperature. So when you take a bath, you're trying to do the bath, you're warmed by your body, it's too hot. Actually, it's a relative change in the temperature to what you are to what the water temperature is.
Yeah.
What does that experiment tell us? It doesn't necessarily tell us that that's a threatening temperature or a temperature at which death might occur, but it tells us about the relative change, does it not?
Yes. So I think it's a sudden change of temperature. I still think there are lots of questions we need to answer about the experiment, which is why I'm sort of throwing the study at you. I think it's still something we're fully digesting as to how we're going to frame it and how we feel it's placed in the literature. Because there is a very interesting set of experiments of frogs, aren't there, when you slowly bring it up to temperature and they won't necessarily respond even when you expose them to quite high temperatures. So it's a good question. I would say, I think it was 50 degrees C, that's quite a high temperature, so it's significant, and we did find that it was an equivocal response, at least in Grammostola spiders, to a mammalian model. So that answers the question.
Yeah.
Good.
Any other questions for any of our speakers from this session?
There's one question. From
Oh, there's a
David
question
Bishop.
online. Okay, there we go. How does 52 days compare to something like a mouse?
I'm really sorry, David. I don't know that off the top of my head. I would have to check. To what extent is that a response? Is that learning or is it physiological? I think how it compares to a mouse, I would have to check the literature. I don't know off the top of my head. I'm really sorry. Good question, though.
Great, yeah, great. So there's another one for Katie. Okay, do you know if the paint was taken off by the insect painted or by its companions?
sorry.
So that's for, here we go, from Lee. Oh, there's another one for you. You've got two, Katie. Well, you can start with the bottom one. Do you know if the paint was taken off by the insect or by its companions? That'd be the first one.
That would be quite a hard question to answer, I think. I think the main, to be honest, I think some of the things that affected how much it rubbed off was the application method. I found when I used the pen to mark the cockroaches, it was, I think one of the reasons why it was so successful compared to some of the others was because like the application method, drawing straight on with like the nib of a pen, it dried like nearly instantaneously, whereas some of the toothpick method ones, they took a bit longer to dry. So I think some of it was they went straight into the enclosure that I put them into to live in for their time period. And it rubbed off like straight away because it wouldn't dry quick enough. So that was, I think, one of the ways was the application method that affected how well it stayed on.
Great. And you had one more. Any work on mortality in the field as a result of marking, e.g. increased risk from predators? Do you think the marking might increase the risk of predators?
So that wasn't really my study area, but I think that really would be an interesting area to study. Mine was more marking them to use in studies so we can identify them from one another. But that would be really interesting, I think. I think it would possibly, depending on the colour you used and the place you drew the marking on, I think it could definitely increase the risk of predation.
Great. Any more questions? I had one for David. It's really, really interesting study that you described. What's your kind of next steps with this experiment? Where are you taking this research next?
Great question. So this welfare stuff is I think fascinating because there's just so many things we don't know. So there's an enormous number of different ways we can take this. So I'm quite keen to explore how their social environment might impact their welfare. So I'm interested in social interactions in general, and so understanding how gregarious species might actually be in a better welfare state in large groups versus small groups, versus isolation and so on. I think could be really interesting because then that might allow us to make general recommendations for these species that have a natural history where they're commonly found in groups would then prefer to be in groups in the lab versus something like the tarantula, which might be found solitary in the wild, might then be better off kept alone in the lab. So that all seems relatively straightforward, but getting nice empirical evidence behind with some welfare indicators, I think would be really cool.
Brilliant. Well, thank you very much. And thank you to all of our speakers for this session. Another round of applause for you. Alrighty, great. So I make it time for a little break. If you guys want to have a little break, a little mosey, and then we meet back for our next Let's Talk, which will be Andrew Crump. So brilliant. Thank you all. Well, Megan.
I'm going. Black soldier flies. I don't understand.
For those of you who are online so couldn't see this, there's a lot of black soldier fly talk going on, very intense. We had to break that up in order to get back to the schedule. And so now we're going to be handing on to Andrew, who's going to be talking to us about bumblebees. So over to you.
Cool, so thank you very much everyone, and thank you especially to the organisers, to Eleanor and so on, for giving me the opportunity to speak today. So I guess I have two jobs really. I'm part-time as a lecturer at the Royal Vet College, and also part-time at the London School of Economics, in terms of thank yous to all of them. And I'm going to be talking about motivational trade-offs in bumblebees, and think about whether this is evidence for insect pain. And I think it's fantastic that the Royal Entomological Society are really starting to take this issue seriously and to think seriously about insect welfare and to set up this special interest group. I think that's wonderful to see, but I'm going to start by, I guess, expressing the very opposite sentiment, which is, wouldn't it be nice if we didn't have to worry about insect welfare? If we didn't have to think that insects feel pain? It'd be nice for scientific research, where a lot of really important applied and fundamental research is done on insect models, things like Drosophila. Be nice for agriculture with insect farming sort of burgeoning, to not have to think about the welfare of these livestock like we do for conventional livestock. It'd be nice for legislation, where I've got a picture of the Animal Sentience Act here, which is our most recent piece of animal welfare legislation in this country, doesn't cover insects, and nor does basically any of our other welfare legislation, either here or across the world. Be lovely for using insecticides if we don't have to worry about the welfare of the animals, both the ones that the insecticides mean to kill, and also the ones that they don't mean to kill, the many, many others. Be wonderful for things like malaria eradication programs. I'm sure that anyone here who's tried to get a paper through peer review on insect pain is familiar with that classic reviewer comment of, what about killing malaria mosquitoes? That's a huge potential issue. And of course, just in day-to-day life, we kill huge numbers of insects. So just driving a car or taking a walk or scratching your face, you're going to kill huge numbers of insects and potentially quite painfully. So wouldn't it be nice if we just didn't have to think about this issue? I guess that's summed up best by thinking insect pain would be a massive pain to think about. So ideally, we wouldn't think about this issue. And I think historically, if we're honest with ourselves, that's sort of what the entomology community has done. Either because, we've all got a lot of concerns in our day-to-day life and in our work, just not really thinking about the issue, or because serious arguments have been made against the idea that insects might feel pain. And one of the main arguments against that, I'm very glad that Megan has already discussed this. I think Megan did the same Google image search for the picture. But this idea that nociception and pain are not the same thing. So nociception is just the processing of noxious stimuli, and then pain is the unpleasant feeling, which is sometimes associated with that, but not always. So we can have reflexive responses to noxious stimuli, where nervous impulses go to the spinal cord, and then you have impulses sent back to the muscles, which cause a withdrawal. But that can happen separately to the conscious sensation of pain when those impulses go up to the brain. And so if this can happen in humans, if we know that humans can withdraw or avoid injury without any conscious pain, the argument goes perhaps insects can do something similar. And so that was an argument that was made in this Eisman et al. Paper, which again Megan talked about. And although it's quite an old paper, it's been very influential. It's still getting lots of citations today. And this is one statement from that paper, where they said that mammalian pain incorporates response motivation whilst allowing flexibility of action influenced by the overall sensory context. And this is contrasted by the fact that insects appear to be reliant on relatively rigid programmed avoidance and escape responses. And I still hear this an awful lot from ethicists and from scientists saying, well, sure, insects might withdraw from injury. However, that's a very sort of rigid, inflexible response. And so a kind of argument against that and potential experimental evidence against that, as David's already discussed today, is motivational trade-offs. So this is the question of whether the animal will trade off the motivation to avoid a noxious stimulus against a competing motivation. Because if the animal does this, then the response is not a simple reflex. It must involve centralized decision-making. I think you raised some sensible questions about whether we necessarily need centralized decision-making for pain, but traditionally that's been seen as important. And also that the response is modulated by context. In humans, this is a key hallmark of pain. And so I think what motivational trade-offs do, if the animal is capable of doing them, is not to say, yes, this animal definitely feels pain, but it does make that argument that the animal definitely doesn't feel pain a lot more difficult to make. In other words, it says that pain cannot be easily ruled out. We cannot simply say this animal is a rigid reflex machine that always responds inflexibly, but pain is at least plausible. And if pain is at least plausible, then I think we should start seriously considering the welfare of those animals. So that's, I think, where motivational trade-offs come in, this sort of picture of evidence for pain, evidence for sentience. There's some really influential research by Bob Elwood at Queen's University Belfast and colleagues, particularly on hermit crabs. Hermit crabs, these wonderful crustaceans, they go around in empty snail shells and have really consistent preferences in what kind of snail shells they like to inhabit. So that depends on various things like the size of the crab and the size of the shell, but also different species of shells that hermit crabs will prefer. So hermit crabs absolutely love these Littorina shells. They go absolutely crazy for them. And so if you give them a Littorina shell versus a gibbula shell, they don't like gibbula shells, they'll almost always choose the Littorina shell. And so the really cool thing about this study, and I think it was really paradigm shifting at the time, was to say, okay, if we put little copper wires in those shells and we administer electric shocks to the hermit crabs, so this noxious stimulus, will the shell quality affect the voltage required to cause the hermit crabs to evacuate that shell? What they found was that yes, for those really great Littorina shells, the hermit crabs took this higher voltage in order to evacuate. They were trading off the shell quality, whether it was a Littorina or a gibbula shell, against that noxious stimulus avoiding that shock. So really influential work, and has been, I think, very influential in convincing a lot of people that potentially decapod crustaceans and hermit crabs in particular may well feel pain, that they demonstrate this motivational trade-off. Like I said, it's been quite influential. So my colleagues and I, when we were drawing up a list of criteria for sentience, basically if you sort of tick off this criteria, can the animal do this, 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 and this? If they satisfy all of them, that's a stronger case for sentience. When we were doing that, we included motivational trade-offs. And I think for a lot of people, they're seen as quite a compelling line of evidence that at the very least, the animal is not this kind of rigid reflex machine, that sentience can't be easily ruled out. And so to come on to our study, the question was really, can insects perform nociceptive motivational trade-offs? And I should say at this point, but a big picture of class I didn't forget. This was not my study. I was not the lead author on this. I was just a sort of very unimportant middle author. It was led by Matilda Gibbons, who Megan's already discussed. Fantastic PhD student led this work. She couldn't be here today, unfortunately, because she's off doing a postdoc in America. So fortunate for Tilda, I guess. But the work was basically looking at these two motivations. The noxious motivation was to avoid 50-degree heat, which bumblebees find aversive. And then the positive motivation that was being traded off against that was the motivation to consume high concentration sugar solution. So as I'm sure you guys are aware, feeding on nectar, bees absolutely love sugar water. And the more sugary the water, the better. The bees go absolutely crazy for high concentration sugar water. So that's kind of the positive motivation. This is our setup. You've got the nest box here. And then the important part of the experimental arena is that square bit in the middle. There were 4 feeders arranged in a sort of semicircle. 2 were one color and 2 were the other. So there were 2 pink and then 2 yellow. And those different colors, those conditioned stimuli, were associated with different sorts of settings. This was counterbalanced, but for the sake of simplicity here, I'm going to say that the pink was always 40% sugar. So 40% sugar solution, which is really high, and this noxious 50-degree heat. The alternative, the yellow feeders, they varied between the bees, but they were either 10, 20, 30, or 40% sugar and no heat. So basically what you had is this gradient of difference between these two types of feeders. So in the 40-40 condition, they all had the same high concentration of sugar. But in the 10-40 condition, the 40 was a much higher sugar concentration, but was associated with this 50-degree heat. Now, I've just got a nice little video I could play, because people always love seeing bees. So this is just looking down on those feeders. And the bee basically had to stand on a heat pad, which was at the bottom. And that can be heated or not heated, and they fed on the sugar through these little sawn-off Eppendorf tubes. This is also because a question I always get asked on this is, how on earth do you tell bees apart? We have these little number tags on the top of them, which we glue on. So that's how we do that, to preempt that particular question. So what were the results? And I think you can see here, they're actually really nice, clear results. So at the side of this graph here, we have the proportion of feeding events at the high quality feeders. So those pink feeders, the ones that always dispense the 40% sugar. And then along the bottom, we have the sucrose concentration available at the alternative feeders. So between 10 and 40%. And in terms of sort of the broad trends, what you see is that as the sucrose concentration available at the alternative feeders increases, you get this sharp decrease in the number of feeding events at those high sugar feeders. That's what you'd expect, right? You'd expect for 40-40, broadly speaking, them to feed at them all at the same amount. But when it's like 10-40, you'd expect them to go to the 40 much more. But the interesting thing is what you see when you compare when the feeders were unheated versus when they were heated. Because what you find is that for those heated feeders, when the feeders were heated, you actually get a bigger trend, a stronger trend. So this is showing us that, yes, the bees were avoiding this 50-degree heat. And yes, they were making a trade-off between heat avoidance and motivation for high concentration sugar. In other words, they were putting up with that 50-degree noxious heat if there was this massive disparity in sugar solution, but they weren't if it was more even. So they are making this kind of nociceptive motivational trade-off. These results are based on when the sugar water and the heat were present. We thought that the bees were making the decision before they were able to sense that, but just to demonstrate that they were, we did a memory test. So this is where we simply used water and we had the heaters turned off. You can see essentially that the results persisted for the memory test. So the bees were showing basically no difference between the heated condition and the memory test, but they were showing a difference with this unheated condition. So yes, the results persisted the memory test. Yes, the bees were doing this based on these conditioned stimuli, these color stimuli. And so the kind of take-home message is, what does this show? Well, firstly, I think bumblebee heat avoidance isn't a simple nociceptive reflex. So these kind of Eisman et al. Arguments, which are still widely cited today, don't really hold water. I think we can say that with a fair amount of confidence now. The memory test also reveals that there's centralized information processing. So in those early hermit crab motivational trade-off studies, and in our original work, there was direct sensory access to all the stimuli. And we now know from work on, say, C. elegans, that very simple neural mechanisms can actually achieve those kind of trade-offs. Whereas here, what we see is that the animals are using conditioned stimuli, they're using memories of those stimuli in order to make the trade-offs, which potentially is more complicated and reveals centralized information processing. So I do think, as someone who works with insects and sometimes does some not very nice things to them, it would be fantastic if we could ethically ignore insect welfare. That would be wonderful. That would save us all an awful lot of worry. However, I don't think we can. I think we've now gone beyond, with the evidence that we have, the stage of saying we can easily rule out insect pain. At the very least, I think we have to admit that it's plausible and that we should be thinking about insect welfare. So I'd just like to say thank you all for listening. Thank you to all of the collaborators on this, especially to Tilda, who led it, to our funders, and to all of you for listening.
Thank you for a brilliant talk. And we have about 5 minutes now for questions before our next speaker. Does anybody in the, I'll start with anybody in the room have any questions? Yes.
I was just wondering, because both yourself and Megan brought up the Iceland paper and it's still having quite a lot of influence, and how we can go forward combating that influence that it's having.
I think we're sort of already doing that to some extent. I did have a look because I wanted to include sort of, Google Scholar does sort of page latency citations by year. And it is notable, I think, that the Iceland paper has stopped in the last few years getting as many citations. And I think quite a lot of them in the last few years have been us saying we should. So I think to some extent we're now moving beyond that. I think in the last 5 or 10 years, there really has been a lot more work on insect sentience, pain, welfare, where really there was next to nothing before. And so I think, yeah, just sort of making that point. Often now I see that people are citing it who aren't necessarily from entomology or even from insect pain. So there's a lot of ethicists still citing the paper. There's a lot of more general animal welfare scientists, for example, citing it. So just sort of reaching out to those communities. So it's fantastic you've organised such an interdisciplinary meeting. Yeah, if you have any other ideas for how to get out the word on this, that'd be fantastic. Yeah, Megan.
I'm going to be extremely rude and also answer this question. You should cite the Gibbons paper, because that is now the best, most up-to-date information on insect pain over the Iceland paper. Also, Tilda and I and a couple of other folks are working on a paper called The Era After Iceman, Insect Pain in the 21st Century. And if that paper passes through peer review, because we all are worried about mosquitoes and malaria, I would be jazzed to see that kind of paper also cited as a direct response to why we need to move past the past of insect pain.
Absolutely, and where there have been more recent sort of reviews critical of insect pain, I think there are some good arguments why, this is not a slam dunk by any stretch. But I think, as Megan sort of suggested earlier, reviews that came out in sort of 2016 and so on, even more recent ones, are already, I think, being debunked by experimental evidence. There's an awful lot of kind of, we haven't shown this in insects yet, and then of course it's very easy to actually show it. And so I think that's one of the real benefits of that Gibbons et al. Paper that we worked on, is that you sort of highlight these big evidence gaps and say, look, we need more research, rather than saying insects just can't do this. But yeah, thank you.
Yeah, great question, great answer. We do have time for one more question, probably. Does anybody else? So what about online? Do we have any questions from our online? Oh, random factoids. Bees have around 1 million neurons. There we go. We get a bonus one.
Depends on the bee.
There we go. Depends on the bee. Yeah, it doesn't look like there's any more questions there. Alrighty. Well, thank you very much. We'll round it up. Do we have the piggy bin?
I just.
Oh, perfect.
Yes, we do enough.
There you go.
Thank you very much.
Alrighty. Well, over to you when you're ready.
Thank you.
And if you could say it in front of that banner-ish, just for the microphone, for the benefit of people online, that would be great. Thank you.
Okay, I'll stand here. Hi, my name is Andrea. I am a postdoc at the Francis Crick Institute in the lab of Lucia Pietro-Godino, and I'm coming at this whole insect welfare question from a completely different perspective, or maybe not so completely different, but from a perspective of optimizing experimental design. So I'm going to be talking about a story that I worked on together with my collaborators, Lee Haynes and Alvaro Acosta-Serrano at the Liverpool School of Tropical Medicine. And this is about pregnant flies, which I will tell you about in a minute. So I work with tsetse flies, and you may have heard of them because they are obligate blood feeders. They feed on mammals mostly,
And while they feed, they transmit trypanosomes, which are parasites that cause sleeping sickness in humans, and the equivalent disease Nagana in livestock. So they are endemic to sub-Saharan Africa. They have spread to pretty much all major habitats in that region, and they feed on a variety of different hosts, as you can see. And as you can imagine, they're not only a problem for public health because they spread sleeping sickness, but also they have a major impact on economic development in the region, because if they infect livestock, the livestock is unlikely to survive, and so it actually inhibits agriculture and farming in those regions. But that is not actually why I'm interested in them. I think these flies are really cool because they give live birth. So here we have a tsetse mother that is very pregnant. You can see that the larva fills the entire abdomen here, and she's about ready to give birth. So this larva is now third instar. She has actually nursed this larva on something that is akin to mammalian milk, and we call it tsetse milk. It is secreted from modified accessory glands directly into the uterus, and the larva eats it there. And then when the larva is born, it is essentially as big as its mother, which is pretty astonishing coming from a human perspective. So this larva isn't going to feed once it's born. It's just going to dig down into the ground and pupate. And then pupal development obviously depends on a variety of factors. That could be soil temperature, soil humidity. But really the larva doesn't have much choice. It'll just dig down and pupate. So the choice of where this larva is going to pupate depends very much on where the mother deposited it. So how do these mothers then choose suitable sites? So over the last 50 years or so, there was quite a lot of work done in the lab that suggested that these flies have a pheromone where the larvae pupate. As they pupate, they release a pheromone, and the pheromone then attracts females that come in and deposit their larvae at the same site. And that would obviously be great news for vector control strategies, for example. We could use that pheromone to attract specifically pregnant females and trap them. But unfortunately, the situation in the field is a little bit different. So in the field, it's not just larvae in the soil. There's all sorts of microorganisms, bacteria that all release some sort of odors. The soil is often covered in leaf litter or other organic debris. And so there's a lot of things going on, and they all release odors that contribute to this really complex odor bouquet. And the pheromone may or may not get lost in that. And in fact, when a recent field study tested this pheromone in the field, they couldn't find any attractive effect at all on tsetse mothers. So that's a pretty substantial gap between lab results and field results. And so one of the motivations for our study was to work out whether we can bring our lab experiments closer to what is required to actually be able to get relevant results that are relevant to the field. And what we have to think about then is our pregnant mothers, right? Because if they're stressed, they very clearly show us that by aborting their larva. And so in the study, we did quite a lot of things that I'm going to talk about in a minute to reduce stress for our flies. There's many different things that may stress our flies. One of them is obviously low quality food. That's something that we can very easily control in the lab. Other things might be too low or too high temperatures that we expose those flies to. Bad smells, especially in the case of this fly, Glossina morsitans, they really don't like the smell of humans. So they may abort their larva if there's a human in the room. And also the wrong light regimes, because obviously we're talking about a very specific behavior here that is larviposition. And when they larviposit, they don't tend to fly out into the savanna to host seek. So bright open phages are usually connected with host seeking, whereas this behavior in particular, they will usually sit in the shade under tree branches before they larviposit. So bright open environments are an absolute no-no for these in this behavioral context. And so what we did to minimize all these stressors is obviously we have to cool them down in order to select them before experiments, but we kept that to a minimal time, usually less than 10 minutes, and the flies recovered really well, and we had very, very few abortions in the time after cooling them down. We banned humans from the insectary during the peak larviposition period, which is between like 2 and 6 p.m. in our setting. so that we wouldn't interrupt our mothers giving birth. And we actually stained our cages brown. We used black tea as sort of a natural stain to stain these cages to just give them a slightly dimmer light environment. And then we measured everything that we could to confirm that our females weren't stressed. So we tested over 1,600 females and we got over 1,400 larvae from them. And that translates into a larvae position rate of over 85%, which is really good. And it's also really consistent across our experimental conditions, which don't matter right now, but I'll get back to them later. Our abortion rate, crucially, was really low. So it was less than 0.5%. And that was really encouraging to see that. And the same way, the mortality rate among females was less than 2%. So that was also really good. So this refers to the mortality during the experiment in our experimental cages. And then, of course, we can look at the larva as well. So in this case, if we look at larval health, what we looked at was the weight of the pupa after it was born. And so here we have our 3 different conditions, and they're compared to the pupae that we found in the colony at the same time of the experiment as a control group. And what you do see is that all of our experimental groups are slightly smaller than the control group, but actually that doesn't worry us too much because we used a lot of first-time mothers, and generally the first larva that they birth is going to be slightly smaller than the subsequent larvae. So this is actually completely expected, and what is encouraging about this is that our pupal weight distributions fall well within our colony distributions. So from all of this, we essentially conclude that our mothers and our larvae were fairly happy and stress-free. And that was a really nice result. We were very happy with this. And we then, of course, expect that any results that they show us is something that we can trust. Except that, of course, at this point, our stimulus is still pretty artificial. So what people have done before is they have used white sand in these kind of containers. And in the sand, they put like the pheromone or some sort of stimulus and make the females choose. But the natural situation in the field looks a bit more like this, right? So you get like dark soil covered in leaf litter, and generally, as I mentioned before, the flies don't actually deposit very much on exposed soil. They tend to deposit in underground burrows, like warthog burrows, or under fallen logs in these kind of areas. So this is not very natural for them. So what we then did was to give them a choice between 2 slightly more naturalistic substrates to larviposit on. One was natural color sand, and then the other is leaf litter. And both those stimuli were autoclaved and then dried very thoroughly to make sure that they don't emit any volatiles that might affect the flies. And then we put our 50 flies into a cage with those 2 stimuli. We left them there for 48 hours, counted the pupae that we got in each of the stimulus trays, and then analyzed the data in this way, where we calculated a preference index, where we took the number of pupae that we found in the leaf litter minus the number of pupae that we found in the sand, divided that by the total number of pupae. And so the preference index, if all pupae are in leaf litter, would be 1. If all pupae are in sand, it would be -1, and 0 if it's 50-50 and the females don't have a preference. So in this case, what we found was a preference index of about 0.8, which is really good. It's a really strong preference for the leaf litter. They really did not like the sand. Every single study before this one used sand as their larviposition substrate. Just saying. So we had 90.46% larvipositing on leaf litter. And that meant then going forward, we use leaf litter as our substrate for our subsequent experiments. And just to very quickly now talk about the pheromone, obviously we tested that then, and we tested in 2 different ways. So we had one experiment where we had the pheromone compound pipetted onto filter paper and presented that under a layer of leaf litter compared to a control stimulus that was just the solvent. And then we made it even more naturalistic, and we actually let larvae pupate in sand, and then covered that in leaf litter and compared that to sand in which no larvae had pupated. And again, we put our flies in the cages, and what we found was that they really didn't care. They didn't care in either of those cases. It was essentially a 50-50 random choice. Okay, so where does this leave us then? I hope that I've convinced you today that our flies really weren't stressed and that they really preferred leaf litter. And that makes it reasonable to believe them when they tell us that they don't really care for the pheromone, if there is a pheromone. But I think there are some take-home messages also about experimental design, because essentially what I think is that if we want to see real behaviors that apply to what's going on in the actual field, we need happy animals, and by happy I mean stress-free. And to get there, I think there are definitely two things that we need to take into account. One is an appropriate behavioral context, and that may involve considering naturalistic conditions. And the other one is that we really need to look at stress-reducing measures, and obviously these will be dependent on the insect that you work with, and we should be reporting these as well, I think. But if we do that, then I think we're much more likely not to measure lab artifacts and rather measure something that's actually a real behavior that applies to the insect's natural environment. Okay, these are all the people that were involved in the study. We published this last month in Proceedings of the Royal Society of Bees, so if you want to have a look, feel free. Thank you for your attention.
So thank you very much for your talk. I think this is a really, really important aspect. So I'm really glad you were able to come and talk to us today about, it's not just a welfare issue, it's an experimental design issue. So I'm really glad that you're able to share this with us today. So thank you.
Thank you.
Does anybody have some questions? Anybody got some questions? Yes?
Do you think there's maybe the room for labs to consult with maybe zoos or private collectors, things about ideas for husbandry, things you could test in the lab to come up with these sort of more naturalistic conditions?
Oh yeah, for sure, for sure. What I find in my research is really important to know the natural environment, to have seen it, where those insects live, right? Before I worked with moths, and I was very familiar with that natural context. Sub-Saharan Africa is very far away, and during the pandemic, we didn't get a chance to go. So it is a lot harder. And so it is important to have these resources, these people to talk to who can give like relevant input to how these experiments should be designed.
Any other questions? I've got one. Out of curiosity, how widespread do you think those kind of issues are, like you described about kind of measuring lab anomalies? Do you think that's quite widespread? Is it species specific, or do you think it's a bit wider than that?
So I'm not sure how widespread it is. I think that if you look at a neuroscience context, people have for a very long time acknowledged that you need your animals to be stress-free, otherwise you won't be observing real behaviors. So if it's a neuroscience experiment, I tend to trust it a bit more because I think people are a bit more careful about this. But like in the tsetse field, for example, a lot of it is vector biology, right? And I don't, in this particular case, people just, I think, didn't consider these issues very much. So yeah, it may be field dependent, but yeah, I'm not sure.
Do we have any questions online? Anybody online have some questions? Oh, we've got another question in first, Kate. We'll do you first and then.
So I wonder, you have clearly the capacity to have stress-free and maybe more stressed flies because of the knowledge you have on what makes them stressed. Do you think there's any effects on the larva, epigenetically, or their fitness as a result of the stress of the mother during development?
I don't know. I think there may be people who have looked at that. I don't know personally. It is possible because these larvae are going to be a bit smaller. If they make it through the entire pregnancy, they're going to be a bit smaller and result in smaller flies, presumably. I don't know whether that's going to have an epigenetic effect.
We have one online from David Fisher. Do the larvae develop or survive less well in sand versus leaf litter?
As far as from what I've seen, it doesn't really matter to the larva much. As long as it has a dark environment, it will pupate. And I think so. Okay, let me rephrase that. A dark environment, and I think a little bit of physical contact helps. So having some sort of the feeling of being actually burrowed, that helps the larva pupate. But other than that, I don't think there's any developmental differences. Because also under leaf litter, not in our case, of course, but in the wild, they would burrow through the leaf litter into the soil. So they end up in the soil.
We got a very long one from Lizzie.
Not a question.
Exactly, not a question, not a question. Not a question, but thank you so much. It's great to see the use of preference testing with respect to insect welfare. Preference testing can tell us what an animal wants as both a species and as individuals. We can and should be doing this with insects. Thank you very much, Lizzie. Yeah, that's a really, really good point. And David, another question. So why do the mothers prefer leaf litter?
That is a really good question. And one I have thought about quite a bit. I think it is partly because the kind of sites that the females look for are indicated by leaf litter, in the sense that so we're looking for a site where the soil has stable humidity and relatively stable temperature, and leaf litter is a very good buffer for that. And so a leaf litter cover on the ground is a good indicator for a site where those conditions might be given. The other thing is also that leaf litter just simply provides shelter during larvae position. It's a very vulnerable time for them. They are very exposed. The larvae don't have a pupal shell yet, so the larva is very exposed to predators, and that could be parasitic wasps, ants. So a lot of predators on the ground there. So I think that the leaf litter might also provide shelter.
Great. Any more questions? Time for, yeah.
Sorry, I thought of a better one. Did you feel, as a researcher, as in your team, that the team were happier doing this research when they felt they were providing a more natural and better environment for your research subjects?
Oh, good question. Well, certainly in the sense that we were a lot more confident in our results, and like knowing that the animals will most likely give us robust results, and that we can be confident in that is a good feeling, you know. Like, yeah, I don't know. Yeah, that's what I would say. Yeah.
Any more questions online? No, I don't think so. Well, in that case, we've got another short break. There are a lot of breaks. You can tell that I teach undergraduates. I do think. Yeah, but yeah, I do think no one wants to be the speaker before lunch. It's good to have a break. So you're not the speaker before lunch, you're the speaker after the break. So quick break, and then we'll be back for our last speaker before lunch. Alrighty, so we're back, and now our next talk again was something completely different. We've got Zachary who's going to be talking to us about taxi flies. So take it away when you're ready.
Oh, well, not specifically about
Oh,
taxi. No, not
sorry,
taxi.
I was on the last talk. Yes, mosquitoes.
There are taxi in here.
Excellent. There we go.
So, thank you very much. I'm delighted to be here. This is all unpublished data, but if you would like to, if you want to see pictures, feel free to tweet it out. That's my Twitter handle that has one tweet on. And I'm going to talk about this very clickbaity title of "Can an insect-friendly insecticide exist?" So, mea culpa first. This is an insect ethics conference. I kill insects for a living, and I kill these type of insects, kissing bugs. These type of insects, which you've just heard about, tsetse flies. And these ones, which is the Anopheles gambiae mosquito, which is the malaria mosquito. And the reason we want to kill these insects is because they are 3 of the most dangerous animals in the world. And the Anopheles mosquito, which transmits malaria, is responsible for around 600,000 deaths every year. And 95% of those are in children under 5 years old. There's a little bit of disparity there. It says 725,000. That is all of the other diseases that mosquitoes spread: yellow fever, Zika, chikungunya, lymphatic filariasis, etc., etc. But hopefully you can see from these 3 pictures, or videos rather, they have a very specific behavior, and that is that they all blood feed, they're all hematophagous. And that's how they pick up disease and they spread disease. And it leads us to a question: well, other insects don't blood feed, and can blood feeding replace neurotoxic insecticides as a more precise target? So we know that the blood meal contains lots of free amino acids, and one of those free amino acids is tyrosine. And on the left-hand side here is the tyrosine metabolism pathway, and it is detoxified by blood-feeding insects after they've taken a blood meal. And then this enzyme, 4-hydroxypyruvate deoxygenase, which is shortened to HPPD for obvious reasons, when that's inhibited or knocked down, tyrosine accumulates. And then you get excess tyrosine, and you get crystal formation. It's like needles within the body of insects. And we think that's likely to cause insect death. And this work was pioneered by Marcus, who's an Argentinian scientist, and his colleagues in Brazil. And he looked at tyrosine detoxification in arthropods, specifically in kissing bugs. So luckily, we have lots of inhibitors of HPPD. There's 3 classes: the chemicals, the beta-triketones, the diketone nitriles, and the pyrazolones. And for this work, we looked specifically at the beta-triketones, and even more specifically, we looked at nitisinone, which I'll refer to as NTBC. And we chose this drug because it's a drug. It's a human drug that is used to treat congenital defects in that tyrosine metabolism pathway. And we'd already done some work looking at using nitisinone to try and control what's here as African trypanosomiasis in tsetse flies, and we found that when you feed nitisinone to a fly in a blood meal, then the flies die. But when you feed it to bumblebees, the work that Seth did, we found that it doesn't kill bumblebees. So we think it would be quite a good target, a very specific target for blood-feeding insects. This is the pathway for the transmission of malaria specifically. These are the mosquitoes traveling around, laying eggs, mating, and then biting. And of these mosquitoes, they bite indoors mainly, and they bite during the night. And so the majority, the heavy lifting of the reduction in malaria cases has been done through bed nets, insecticide-treated bed nets, and also spraying walls in a process known as indoor residual spraying. But we're specifically looking at this behavior. So once a mosquito is taking a blood meal, it will then rest on the wall, and that wall is normally treated with some sort of insecticide. And so we were thinking, well, could you use nitisinone? The mosquito touches the wall, it's coated in nitisinone, does the mosquito then die? But then other insects that might be in and around the house, they wouldn't be affected. And to test this, we used 3 different methodologies. So firstly, we use this topical methodology whereby you have your insecticide solution, you apply it directly to the thorax of the mosquito. Secondly, we use what's known as a tarsal assay, where you have a little glass plate. Your mosquitoes go and sit inside for 30 minutes. It's coated with your insecticide, and then you measure mortality. And then finally, this bottle assay, which is a modified version from the Centers for Disease Control, the CDC, where you put more mosquitoes, 25, and you leave them in there for an hour. And there's lots of research. These are all sort of standard techniques that are used in insecticide testing. And we're very lucky at LSTM. We have a spin-off called the Local Insect Testing Establishment, and they have what's known as Good Laboratory Practice, which is or GLP. It's a legal standard it's similar to good manufacturing practice, which are the practices that are required for people to make drugs. So we have very high sort of standards for care, and essentially you need happy, healthy mosquitoes to then try and kill them, because otherwise you're sort of running into problems. But specifically for this work, oh gosh, it doesn't normally look like that. So specifically for this work, we blood fed our mosquitoes. So we blood feed them artificially. We have blood that is donated. It comes from the NHS blood bank, and they're allowed to feed at will, and they do like feeding. So the first question was, how potent is nitisinone when it's applied to the mosquito thorax? So we already had an idea that if you just give them the blood meal, they'll die. But what about how does it go through the thorax and into where it needs to be to kill? So this was the topical assay. So we used this insecticide cephal strain, Kasumu, came out of Kenya in the 1970s, and it's now a very well-established colony. And we use this 80% mortality threshold. This is a World Health Organization standard. So for an insecticide to be worthwhile, you have to have killing over 80% at 24 hours. And you can see here, these are concentrations in percentages, and it's your average corrected mortality. Oh no, sorry, this is just average mortality. And we have sort of high mortality over 0.01%. And when we compare that to other neurotoxic insecticides, or rather pyrethroids and DDT, these results are comparable. So we thought this is, onto a winner. So the second thing was, well, what about if you have sugar-fed mosquitoes and you have blood-fed mosquitoes and you put them on a plate that's got the nitisinone on, what happens? And this is our kind of insect-friendly bit. And you can see here you've got your sugar-feds here and your blood-feds, and you see when they're blood-fed, they have 100% killing straight away after 24 hours. And the sugar-fed mosquitoes, they're quite, they're quite healthy, not healthy. They've been through a thing, but they're not dying. There's no real mortality there. And that's basically this. So unfed mosquitoes don't die, but when you have a blood meal, they do die. But what is the lowest dose that quickly kills mosquitoes? So again, using the Kasumu strain and this 80% threshold, we have different concentrations of insecticide and nitisinone on the glass plate. And you see here, over time, mortality increases, and it's always above this 80% threshold of 25 mg per meter squared. And that's good because killing between 5 and 25 mg per meter squared is comparable to neurotoxic insecticides. So it holds up quite well against those broad spectrum insecticides. And to better demonstrate that, we use the bottle assay. So here we were trying to work out the discriminating dose. So we take an LD50 and an LD95, and then the discriminating dose is calculated as being 3 times the LD95. And when we compare that to other insecticides, you see here, this is clothianidin, that is a neonicotinoid. And at the bottom here, you have tolfenpyride as a new class of insecticides that disrupt insect cellular metabolism. And nitisinone fits. Oh, we have gone out. So nitisinone fits just here in the gold.
To make sure it's sharing.
So that's nitisinone. So it's sort of it's comparable to those other neurotoxic insecticides. Now the big problem with vector control currently is that nature has found a way. It's fought back, as it always does, and we have very high levels of insecticide resistance. So in the countries where the majority of deaths are happening from malaria, there's 5 countries. One of them, for example, is Burkina Faso, and they have very, very high levels of resistance to the traditional insecticides. So we want to know, we use these 2 strains, Tiacele and VK7. They have slightly different resistant profiles. Tiacele is metabolic, and VK7 is cuticular. VK7 comes from Burkina Faso. And what you can see here is, although there's a sort of, in Tiacele, there's no real difference between what you saw in the susceptible strain. There's a slight decrease slight increase in the time it takes to kill in VK7. But when you put them together, after 3 days post-exposure, we've got Kasumu, Tiacele, VK7, and these are the doses increasing. Interestingly, VK7, for some reason, we're not quite sure why, at the lowest dose, had significantly more depth. Here, this is the sort of mid dose, the 5 mg per meter squared. There was no real difference between them. And then for the 25, and then the very highest, 125, there was no difference. So this this sort of means that our susceptible and our insecticide resistant mosquitoes, they share the same sort of mortality with nitisinone. So future work. So we've looked at the mosquito flies in, it takes the blood meal, it evades the insecticide treated net somehow, or the persons are under a net. It has the blood meal and then it goes and sits on the wall. But what can also happen is the mosquito comes in, it has a little rest first, it looks around, it decides, I like the look of you, I'm going to bite you. So what happens if the mosquito lands and then it takes the nitisinone up? So we have done some work on this with the sumo. It looks like there is no difference whether the mosquito picks up the nitisinone first before the blood meal or whether it picks it up afterwards. It's a lot of work though, because we're trying to reduce the number of mosquitoes that we're using. And these are my two colleagues, Lee Haynes and Claire Rose. And so what we have to do is we expose our mosquitoes, and then Lee is encouraging the mosquitoes to blood feed through heat and through smell. And then Claire has to remove all of the non-fed mosquitoes. So that is a way of reducing the number of mosquitoes that we need. We have to have more staff, so it takes longer to do. The next step would be to see whether it can be formulated. So all of this work is just lab. It's lab work where we're dissolving our nitisinone in acetone and we're putting it on a plate. Chemically, that presents some problems. So we would need to know whether the nitisinone can be formulated to be more effective on walls or surfaces. And then finally, I've spoken specifically about Anopheles gambiae. There are many other species of Anopheles that give out malaria, Anopheles fenestus, Anopheles arabiensis, Anopheles stevensi, etc. And they might have slightly different blood feeding patterns. And then what about UK vectors? So this is quite a sort of insect-friendly tool. So these fleas, ticks, bedbugs, sailflies, midges, could you kill these using nitisinone? So then finally, can an insect-friendly insecticide exist? So maybe, I kind of skipped over it when I was talking about bumblebees, but mortality is not equal sublethality. So what Seth didn't do was he didn't look at the work of fecundity. So more work would be needed, but obviously you want funders to give you more money, so give them something extra to think about. And this was the team, Lee Haynes, who seems to be popping up quite a lot in these things, as does Claire and Alvaro, and George and Rosemary. And finally, any questions?
And thanks again for brilliant talk, and again, a really, really nice, a very different perspective on this very important issue. So thank you. I'm so glad that you could come and chat to us today. Thank you very much. Do we have any questions from the room? Yes, go for it.
I heard the week on a science podcast that scientists working on their microwaving insecticides to get into crystalline structures. I mean, does heating or microwaving help in any way to be more effective, quicker death, in your opinion?
Yeah, well, it would do, because so the size of the crystal matters. So if you imagine in a net, you have the threads, like a thread of cotton, and in those threads are crystals and insecticide. So the smaller the crystal, the easier it is for the mosquito to pick it up through the cuticle. But if it's big, then you could have something that's really, really lethal. But if if it can't pass through the cuticle, it's never going to kill. So there is an infinite amount of work being done on trying to make better bed nets. One of the reasons why malaria has dropped, it's not just that the nets are coated with insecticides, it's that the nets can be washed up to 20 times and they still maintain a lethal dose. And that means in countries such as Burkina Faso, where you don't have as good an infrastructure as you would hope, you can sort of leave the nets and then come back later on, so you're not having to constantly go out and change them. But yeah, it's definitely important work.
Excellent. Whether we reverse some of the resistant strains where there have can work almost fairly simply without you, because I think they used deltametry, didn't they?
Yes.
So it suggests that you can actually reverse.
So because you have to use pyrethroids because they are human friendly. So if you have someone sleeping under a net every night and for every day of the year, it has to be something that's not going to hit them. There's a lot of work going on at LSTM looking at behaviour around the bed net. So we film the mosquitoes and we look at where they're landing, and some of that work has indicated that the mosquitoes are coming in at the top. The top isn't in contact with humans. So there are net designs now where you can have a different type of insecticide that might be more dangerous to humans because it's up at the top of the net. So it's not just, all the research is not just constantly trying to kill things. There's a lot of thinking about how it's best to target them.
Yes, you got a question.
So between them ingesting the insecticide
Yes.
and biting someone, can they still transmit malaria?
So if they die quickly enough, they wouldn't. So a mosquito, when it's the sort of adage is an old mosquito is a dangerous mosquito, because a mosquito has to bite someone. If that person's got the plasmodium, they then have to bite someone else to pass it on. But with the work we've done, we've looked in blood meals as well, and it kills them so rapidly that they wouldn't be able to transmit. Even the break in transmission is important, but also the female has to take the blood meal to lay the eggs. So you would lower the number of mosquitoes.
Any other questions? Do you have questions?
I was just going to ask, do you have any expectation that they might evolve resistance to this new method of fermentation?
You can never say no. Some of the work on the taxi paper, we looked at P450s, cytochrome P450, which is an important metabolic enzyme for detoxifying insecticides, and we found that there wasn't. It didn't seem that there would be that sort of metabolic resistance buildup. HPPD itself is a very well conserved enzyme, so the likelihood of it's you can never say never.
Any other questions in the room? No. Do we have any online questions?
We do.
Oh, good. Okay, we've got a couple of online questions. So we had one from David Fisher. Is blood the only way that tyrosine can get to high levels? Wondering if there are any non-target species with unusual but non-blood diets that would be affected.
So we have been looking at, because the male mosquitoes, they don't take a blood meal, and some of the work we did was looking at giving mosquitoes, the male mosquitoes, tyrosine in the blood meal with nitisinone. But that work is ongoing to see, but there is potential that you could have, but they would have to be constantly landing on the surface and then having sort of a high protein diet.
Brilliant. And Lizzie says, apologies if I missed this, but how quickly does the insecticide kill the mosquito?
So at the top dose, it's within 24 hours, and the next dose down, you pass that 80% threshold. The 80% threshold is a resistance threshold. If you have insecticides that are working below that threshold, then the risk of insecticides mosquitoes becoming resistant to insecticides increases because you're giving a sublethal dose. But it's definitely within 24 hours.
Great. Any more questions from the room? No? Well, again, thank you very much. Really great talk. I'm sure you've all been waiting. Oh, it's lunchtime. So do feel free to go grab lunch. For those of us who are not in the room, do grab lunch and do come back after the break. Right, we're free. Great. All right, so ready to go again?
Yes.
So thank you very much for all coming back after lunch. And as I said, do feel free to help yourself to any more food as the session progresses. But so we've heard some brilliant talks already about a variety of different things, but the next couple of talks are going to be a little bit different, and are going to be a bit more focused on the education side of this very important issue. So come to us. So my talk in particular, so this is something that I feel very strongly about, and it's something that I've been trying to kind of work on. And in fact, we are working on a chapter at the moment about this, so I'd love to get your guys' feedback and ideas about this. So the question is, how can we support student decision making about invertebrate welfare? So this is my goal aim. I'm sure some of my students in the audience will be very aware of this. My objective whenever I go into a classroom is for the students to come out and be like, wow, I didn't realize bugs could be this cool. I mean, obviously we're teaching the important things about conservation, genetics, all that kind of thing, but ultimately this is our goal. And as part of that, hopefully, some students, through realizing bugs are really cool, might decide to go on and do a dissertation project to pursue this further, to do a research project, which is great, which is brilliant, budding entomologists, we love it. But this is hard. This is really hard. We are all in this room because insect ethics and welfare is challenging. It's fascinating. It's multidisciplinary, and it's consistently changing. All of these papers are published since 2019, and you can imagine if most of you guys have been working in this area for quite a few years now. There is a lot of literature now on this area. There's a lot of kind of ethical quagmires. There's a lot of gaps in our knowledge about the best way to do things. It's really difficult for us to expect an undergraduate to suddenly be able to get on top of all of this literature and to be able to come up with a research plan, usually in the space of about 2 weeks when they're writing their research proposal while they're trying to hand in everything else. This is challenging. And what can we do to try and support them being able to do this? So on one hand, we have our kind of support. So we have our published literature, some of which, you guys have been presenting today, really important. We have our best practice guidelines, like Megan has been talking about, really, really important. In an ideal situation, you have your support from a supervisor. So this is the key relationship here. In an ideal situation, you have a very supportive supervisor who can very gently take you through things and guide you in your ethical decision making. However, unfortunately, this is not always a kind of ideal scenario, and also we do have some challenges. In many cases, you can have gaps in supervision. In an ideal situation, we know that a student will be being supervised by a dissertation supervisor who is a specialist in what their project is. However, we often know that students might be interested in pursuing a project that's on the edges of a supervisor's kind of interests or knowledge. So there might be some gaps. And also, you do have situations where sometimes supervision changes, maybe there's not as much supervision support as there needs to be. This is something that we've been talking about a lot today, uncertainty about best practices. Sometimes we just don't know what the right thing is to do, which is why the research everybody's doing here is so important, to try and allow us to get to a place where we understand what are the best practices. And also, unfortunately, it's really hard to stand up to your boss. I mean, I'm sure everybody, whether it's a supervisor or a PI, it can be very difficult to challenge some of the assumptions. And actually, this is a really, really important one. We want students to be in a situation in which they feel comfortable making the decisions, to be part of that decision-making, journey, and to really know what they're doing and why they're doing it. So we've got to kind of try and get around this as well. And, I think too, it goes beyond, we can't get through this by just having a session on insect ethics and welfare. That's not going to be enough. It's a huge topic. And we need to think very carefully about how we can integrate this into different levels of the degree program. So to start with, and I'm going to talk about each of these in turn, we need to think about our content and delivery. We need to then think about the course structure and how we can build different learning points along the way. And then finally, a little bit, we can think about the supervision as well. So starting with teaching content and delivery, two things which are really important is showing best practice in teaching, and secondly, communicating uncertainty. So, the old adage, do what I say, not what I do. Unfortunately, we can't do that. There is a lot of evidence, particularly from kind of medical best practice, and this is something I'm going to draw a lot from. If you Google uncertainty or teaching uncertainty to entomologists, you will find nothing. But we can draw from a wide variety of literature from other fields, particularly things like medicine, who deal with teaching students uncertainty all the time. So we know from kind of other fields that reflective imitation is a key learning tool. People pick up things like kind of ethics and kind of welfare standards from watching people do different things. So we have to think about this in our own practice. And so a little case study of a practical that we do at Writtle, many of you in the room might have been part of this practical at some point, is pond dipping practical. I'm sure this is carried out in many different ways at different universities, but we can think about how we might be being perceived and how we can get this across to students. So starting at the top, one of the key things that we do is before we go anywhere near a bug, we spend a lot of time educating about the species, showing them how important and exciting these species are. We do try and chat about the ethical choices made, particularly with the reduction of death. So in our particular pond dipping practical, we're able to take the animals in, we observe their behaviours, we get students to identify them before letting them go. And we then talk about the disposal of animals. And I think this is really important. One thing that we do is make sure that the students who have collected the animals are then responsible for releasing them. And this is something which I think is really important, and we do throughout Writtle. So Katie, for example, to put you on the spot, she did a great job of looking after her own specimens when she was doing her work. And I think that's a very important element, that kind of level of kind of ownership over the work. And then also reducing samples taken. So we do a lot of group work, but also these are my rock monsters. I love using these as a teaching and learning aid. In one of the first lectures, students get given one of these, which they're asked to take home. And we use them to teach about things like dichotomous keys, taxonomy, and things like that. So we use a lot of kind of tool use to allow us to do these kind of teaching, which then we can use it to support. There's nothing that we can ever do to replace the field teaching. There's nothing that can replace somebody seeing a dragonfly larvae for the first time. Can anybody remember seeing a dragonfly larvae for the first time? So exciting. You can't replicate that with anything except for seeing it in real life. But we can use a lot of additional tools to kind of reduce what we might be working with. Communicating uncertainty. So this, I think, is a really important thing that we need to explore a little bit more, particularly if we're trying to teach in this field. You know, as we've been talking about, there's a lot of uncertainty about the ethics of things we should be using, the best practices, even things like kind of euthanasia. And, you know, students will be, you know, in an ideal situation, students will be encouraged to write their own research proposal, which will be guided, but they will have a role in deciding the types of practices that are used. So they will be dealing with this uncertainty, and you know, what can we learn from other fields to help us support students in a good way to overcome this? So this particular paper here, which is really good, again drawing from the medical literature, students react to uncertainty in different ways. So these are all different words that students have described when talking about uncertainty. Nobody likes to feel uncertain. I don't like feeling uncertain. If I don't know the answer, it's stressful. You feel that kind of degree of stress. But the interesting thing is they found that students often will react in quite different ways. So on one hand, you have certain individuals who will take it in quite a positive way. They might kind of embrace this idea of what there's no kind of right answer. We can develop this, that, you know, we can justify this, we can develop this area. So they might ask questions. They might let go of perfectionism, which is actually a really important skill. So in general, kind of could be an important learning catalyst. On the other side, you might have a lot of students who react quite negatively. So you have students who might deny uncertainty or avoid questions altogether, or even avoid the situation. And you know, I know I've been quite tempted to do this in certain situations, but ultimately we have to be very aware of the fact that we're putting students in a situation where they have to grapple with this uncertainty, and we have to think about the fact that they are diverse learners and different people will react differently. And we have to make sure our learning environment reflects this kind of diversity that we have. And how can we get over this? Again, oops, oh no, what have I done? This is terrible.
Thank you. I clicked the wrong button, that's what I did. Just make sure. Sorry. Dear. There's too many buttons on this clicker. I usually only have 2 buttons on my clicker.
There we go. Thank you. Great. Okay, so what can we do to get around this? So again, we can draw from the medical literature, but also the veterinary sciences. The vets are much better at this than we are. They talk about this a lot. We can learn from them. They just suggest art-based exercises can be a good idea. So things like creative writing, peer-to-peer discussion, or scaffolding to encourage reflections. And I'm going to talk about both of these in just a little bit more detail later on. Okay, so we've done our teaching and content delivery, but now thinking about course structure. So at Writtle, again, this is a case study for what we've been doing here. There might be better ways to do this, but I thought this would be interesting to share as a case study for what we're trying to do. The first time that, so this is our animal science, animal management students, the first time they interact with insect welfare, it's under husbandry. During this, I introduced them to the concept of insect welfare. We do this in largely kind of a classroom-based setting, but we also have been integrating, so some of you will know, but some of you will not know, but we're setting up an insect farm up at the farm, which will allow us to breed black soldier flies on campus, which is an amazing learning tool. And so through this, so this is a picture from our different husbandry classes, but we get students to think about what they have learned from other species, what they have learned throughout their degree, and what they can then apply to how they would manage an insect farm. If we were to get them to come up with a management plan, what kind of things would they think about? And so there's a lot of kind of discussion here, and welfare often comes up. So it's quite kind of structured. We talk a little bit about different aspects of husbandry. In fact, you can see in the background some of the bugs that we get them to handle. These lectures always completely bananas and very good fun. But this is the first time that it's introduced in quite kind of a gentle way and in the context of insects as feed for other animals. The next step that they experience this is then a little bit more kind of out there, kind of pushing it out into slightly more uncomfortable territory. So we get them to think about cephalopod farming, which obviously is a really good case study to start grappling with those ethical issues. And we do this by having a kind of a debate, but it's quite a structured debate. We get them to kind of go through these different sources and then debate about the kind of pros and cons of this. And then finally, in the final year, so then we start to introduce the even harder levels of kind of debate, particularly looking at the relationship with animals, including invertebrates and people. So looking at the really hard topics like parachute science and things like that. So by the end of their journey, so this happens in first year, this is in third year, and then this is at the end of the third year, we hope that by being experienced and by debating and getting to grips with uncertainty and also insect ethics, they will be in a much better situation by the time they hit the capstone project to have that kind of background of knowledge and that experience of grappling with the uncertainty in this field. So hopefully they'll be more confident in their own ethical decision making. So finally, onto dissertation supervision. So, as I said before, challenging mentors in practice can be very difficult. And we can see again from the medical literature, students really struggle to do this. And, for a very good reason, it can be kind of very detrimental to students who kind of feel like they want to suggest changes. But one of the things that is key is for students to feel confident in their own knowledge in order to feel like they can make those challenges. My suggestion would be that alongside the kind of support at the different levels that we've already discussed, maybe kind of looping back to some of the first discussions, maybe we can give them a framework to help encourage these conversations between supervisors and students, and also to facilitate student self-reflection in this area as well. So this is not to replace the current guidelines that we've been talking about, but instead, I would suggest using kind of some very, very simple questions alongside these guidelines to encourage students to kind of think about these areas in quite a critical way. In particular during collection, husbandry, experimental design, and disposal. So some examples of the very simple framework that we might be using would be collection. How are you sourcing your specimens? And have you addressed any ethical concerns? So very straightforward, not stressful, and hopefully a very kind of a useful tool to encourage students to start to think about that and have those discussions. Sample size, again, very simple. What sample size are you using? Justify your sample size. And again, very straightforward. It's hopefully not going to stress anybody out, but will hopefully be a useful tool to allow students to kind of think about this. Husbandry, how and where will the animals be handled and housed? Again, there might not be a set answer to this. There might be multiple ways, but the key thing is to encourage people to take the time to think about and justify the decisions they've made. And finally, disposal. Again, this kind of metric isn't to make a judgment about the best form of disposal, because if they are working on any number of species, we might not have that information. But the key thing here is to encourage the discussion and to make sure that they think about justifying their methods. So overall, hopefully kind of thinking about this, and as educators, as lecturers who are thinking about kind of building this into our students, hopefully thinking about how we can provide this kind of background and practice can kind of help support them by the time we get to the dissertation level, and also thinking about perhaps this questionnaire-based method can help structure those discussions with supervisors. So hopefully, we're thinking about all these things in tandem. We can think about how hopefully we can kind of move away from this to a kind of a bit more of a tilted pattern like this. Hopefully, we might be able to help overcome some of these gaps in supervision and also some of the difficulties with challenging supervisors, and most importantly, encouraging students to have confidence in their own learning, to have these discussions and feel like they can take direction for their own projects as well, which is ultimately what everybody wants. So there we go. So that's all I've got to talk about today. And I just really wanted to say big thanks to the whole teaching team at Writtle, who's very supportive of trying new, weird, wonderful ways of teaching. Nikki in particular, and Sophia, who you saw her mugshot earlier. She's been helping in the bug barn. She's wonderful. And finally, Refly Cycle and the funding provided by the UK Knowledge Exchange Partnership. So there we go. Does anybody have any questions? Yes?
Are you going to use the black soldier fly to feed the animals in the unit?
This is the plan. So the plan is at the moment that we will be able to use some non-consumption food waste, so kind of carrot tops and that kind of thing, feed it to the insects at the farm, and then put that back into the animals. So not the animals that are going for slaughter, but like particularly focusing on the exotics and things like that at the Titchmarsh Centre. So again, kind of feeding into that circular economy thing, and hoping to have students involved the whole way again as a teaching tool. So yeah, we're very excited to get that up running. We're hoping to get it up running by the end of the month, actually, aren't we? So yeah, we're nearly there. Do you have a question?
You touched on some ethical considerations, but is there any specific ethical considerations for invents for students?
Yeah, so I think that one of the key things that is particularly important is thinking about kind of husbandry and also how you might dispose of the insects. Because, for example, things like, in the case of the pond dipping, it makes a lot of sense to release them back in the wild, but actually in many cases that might not necessarily be ethically the right choice. So some of these kind of questions about, should we release them, should we euthanize them, if we're going to euthanize them, how are we going to euthanize them, then the questions are often not quite as straightforward as you originally see and can often depend on the species. So it takes a lot of kind of thinking through almost, and it is much better if people can kind of come to their own conclusions rather than being kind of told what to do oftentimes. But those would be the two things I would be most interested in to make sure people kind of think through. Yeah. Any other questions? Yes.
Do you have plans for other species in the bug barn?
I think at the moment it's going to be black soldier fly. Maybe because it's taken a really long time for us to get that set up. I would love to see a situation where we had other species in there as well. But I think for now we're just trying to get this started. Better to focus on one species and do it really well. But having said that, though, in Titchmarsh we do have a lot of other species that we use teaching husbandry. So particularly in that module, it's really fun. I start them handling snails. We get more difficult, then it's wood lice, and then it's locusts, or like locusts or crickets, and then finally it's cockroaches, which is always very fun. So we do teach them to handle a lot of these, even if the farming aspect is very focused on that. Any other questions? Yes.
So the model for this to battle with students.
Yes.
I noticed that it's mostly focused on undergrads.
Yes.
Is there any possibility in the future that you, for Writtle and other institutes, to incorporate this into postgrad and even further?
Yes, Absolutely. You might have noticed that some of the things that I talked about for the undergrad, you guys might have received at a kind of a higher level. So I have been trying to integrate it into your master's things as well. But yeah, I definitely think that particularly having the framework at the end to have to talk about these discussions with your supervisor could be something useful for post-grad as well. At the moment, so by way of context for those of you who've not read lights, Writtle doesn't currently. We've got some senior Writtle lights over there. So Writtle, like all other institutes, doesn't require ethical approval for insect-related projects. But however, as the only person who supervises insect projects at the moment, I require my students to put through the paperwork for the Invertebrate project, which means that a lot of the discussions that I had, sorry, I keep picking you, Katie, because you're right here. But you then end up having those discussions with the student during the ethical review process as they start kind of talking about the ethics as applied to the kind of vertebrate structure. But actually, the vertebrate structure isn't really fit for purpose for invertebrate research. Things like collection, where you're getting your animals from, the kind of disposal methods and that kind of thing is very, very different for invertebrates. And then vertebrates, and also we do have the challenge that invertebrates are so kind of multidimensional. We have so many of them and so many different species. I don't think that they lend themselves to a very strict kind of tick boxy type, framework. I think using something like this might lead to kind of more helpful conversations rather than trying to kind of fill in all the boxes to get it reviewed. Yes.
So you were speaking more generally about undergrads struggling to sort of challenge their supervisors or sort of rock the boat as such. I was just wondering, how would you encourage undergrads to
do that? Yeah, so that's a really, really good question. I'm not necessarily saying that all undergrads should fight against their supervisors. That's not what I'm trying to say. Particularly my students who are in the audience. I'm not saying that at all. Because often your supervisors do definitely have the best interests and that kind of thing. But it's more being confident to ask why. I think there can be situations, and I know I've experienced this in my own education, where your kind of supervisor will tell you to do something and you will do it, and then you don't necessarily ask why. And usually supervisors love to be asked why, and and then when it gets to the fact that you're presenting, you'll have that kind of deeper level of understanding of your project. So it's not necessarily that you should go into combat against your supervisor, but it should be a case where you are feeling confident that you can ask why, and you keep asking why. And you can keep asking, you can be that toddler and just but why, and that should be something that we encourage. And I think kind of based on some of the literature in the kind of medical literature, one of the key things is for students to feel like they have that basis of knowledge in order for them to feel confident enough in their own learning that they can then ask those questions. So I think kind of supporting the learning along the way is really key, but also kind of institutionally being aware of this, and also being aware of the fact that you have this diversity of learners and some of whom might react differently as well. So yeah, good question. Yeah.
Sorry, I'm going to piggyback off of that question.
Yeah,
do you think that when we're saying, we ask our supervisors why, do you think that should be the last resort for the student? Do you think the student should be referring to published literature and things first to try and understand, and then ask your supervisor why, because you break through every other sort of information source? Because I know that some supervisors may turn around and say, go and do some reading on it first. So do you think that's sort of a last resort thing? Or?
I think it depends on the context. I think an ideal situation as students, going away and learning about the literature independently and then coming back and having those discussions with your supervisor is really, really important. However, there can be some particular circumstances, the bugs have escaped. You can go and ask your so I think it can depend on the context and where in the project you are.
Yeah, of course, because I'm just thinking of say such like controversial topics, maybe such as disposal. That's quite difficult to read literature on and then talk about afterwards. So maybe that's just from a student perspective, directing how you go about certain topics instead of just being like, why?
Exactly.
Do you see what I mean?
Yeah, and you're totally right. Like I think it's really important for students to be self-directed and to be able to make those, particularly when it's something that's kind of an ethical decision. You know,
Yeah.
it has to be something that you cultivate
Yeah.
yourself. So making sure that you understand the literature and you kind of independently, you know, I'd much rather that a student went and checked out the literature and then came back to me and said, I think they were fine, they're doing this, they're
Yeah,
you know.
this is what I'm thinking. Yeah.
And I don't think, I think most supervisors would be very receptive
Yeah,
to that.
especially if we're looking at a level 6 or a postgrad sort of, if we're integrating like this into a postgrad sort of thing.
Exactly.
But yeah, it's just interesting from a student.
Yeah, definitely. Thank
And
you for
staff.
sharing it.
So,
Yeah, exactly.
yeah.
Yeah. Any other questions? I think you had your hand up slightly good.
This is just like a piggyback off the
Yeah.
last two. If there is too much of a conflict of interest
Yeah.
or too much of a gap in knowledge from that supervisor,
Yeah.
would you be able to swap supervisors?
I mean, ideally, you can work it out with your supervisor. You can work it out with supervisor. Like often at supervisory, you know, dissertations, you have multiple supervisors anyway. So that can be quite useful if you know, perhaps you need a bit more kind of support, or you feel like you need a bit more kind of support in various ways. Having multiple supervisors on a project can be quite useful, and then they can find it out between themselves. So in unfortunate situations in which you can't resolve a conflict with supervisor, there is always the option to change supervisors, but that very, very rarely happens, fortunately. But yeah, but it's always good to kind of talk about, or to talk to your supervisor about. There we go. You had a question.
Yeah, I guess I just wanted to follow up on what you were saying about sort of legislation of insect research potentially.
Yeah.
So I guess the Home Office at the moment are quite seriously considering the idea of decapods in the ASPR, and we could well see that in the next couple of years. Insects very much on the much more long distance horizon.
Yeah.
And I sort of agree with you that the way the ASPR is set up, it would have to be so heavily amended to be possible for insects that it's not really worth doing. So I wondered whether, sort of what would your ideal, if it existed, legislation look like for insect research? Do you think legislation is the way to go? Because just having sort of supervisor discussion seems a little bit.
No, it's a really, really, really good question and a really hard thing to tackle. So thank you for bringing it up. Really, really hard.
Managing uncertainty.
Exactly, and there's a lot of uncertainty here. I'd say it depends on the context. I think within the university context and kind of supervising students, I think one of the things to think about at the moment is we don't necessarily have the knowledge to give really robust, recommendations. Like there's certain species that you can make quite robust recommendations, things like bees, and there are certain guidelines that are coming out, as headed up by Megan. So we are getting these kind of guidelines coming up. I think the challenge is though about the gaps in our knowledge at the moment. So I think that once we have the knowledge to make kind of robust recommendations, then maybe that could be an option. But I think at the stage that we're at the moment, it could be important to leave some kind of leeway. In particular, so we've been kind of talking about whether or not this could be implemented at kind of as something that students have to do with doing Invertebrate projects and the challenge that's come up again and again is the reviewers. The reviewers are often non-specialists, they're specialists in welfare and they're very, very, but how would a reviewer who's a welfare specialist, but not necessarily an entomologist, know whether or not these recommendations for husbandry for this particular species of rare woodlouse would be appropriate over these other recommendations? Like that's quite a big ask for reviewers. So we would need to make sure that we have metrics that they could easily read. So for example, in this, the metric that they would be looking for if we were having this reviewed, it would be to make sure that the decisions are all justified. So we don't necessarily put a judgment on if the student's working with woodlice, they have to keep them like this, but they have to show justification for their work. Does that make sense?
Yeah.
But things might change as research develops. And hopefully it will. And that'll be very exciting. That's why we're all here today, really. Yes.
You've been talking about the regulations in the lab and with students and stuff. What happens when you go commercial and these guys are looking after tens of millions of insects in one big area? I mean, the rules and regulations for that, what you've been talking about, I'm sorry, but just doesn't seem relevant.
Yeah, so it's all, no, that's very fair. But this is entirely focused towards how we're looking after our students in the university. So very, very different to how things might work at the commercial level. So obviously with the commercial level, again, I think Megan would be better placed to answer this than I would, and I'm sure you'll be able to chip in a minute. But my view though, in terms of the that kind of level of production is the fact that changing. And I think the question as an entomological community is the change going to come from within the community? Are we going to kind of lead this change, or is it going to be imposed from outside the entomological community? And I think that's the kind of choice we have at the moment. And so I think it'll be kind of really important to work with industry and to collaborate with industry and policy and ethicists and vets, and to kind of develop the regulations for industry in a very kind of collaborative way to make sure that the really important work that is being done isn't hindered, and also can protect the people who are involved, because I think that is something that is an important part of the kind of ethics procedures. It can protect producers from backlash, from ethical backlash and things like that, to say, oh, actually, we've been following these. So it's not just that it's stopping people from doing something, it's protecting an industry from kind of ethical backlash, which I think is particularly important given the consumer base. A lot of them are very kind of ethically kind of minded people looking for protein alternatives and that kind of thing. So really, it's hopefully going to be a positive thing, but it has to be done in the right way to make sure that it's done in a collaborative way. What's the question?
Yeah,
Good.
I just think you do have to collaborate with those people.
Definitely.
I mean, from my background, when the animal activists were at their height in the sort of 80s and 90s, they are not nice people to be talking to.
No, exactly.
You do not want to be
No,
there.
exactly.
You know, I met them and yeah, they're just, you don't want to be there. You have to do it inside, but you need to get everybody, not just the university people here, but you need to get the commercial farmers in who handle them in big quantities. And they do want to look after their animals, even though they're insects, because everyone that dies is a loss of money, and that's what they're interested in. But you need to get those people on board and positive with you as well.
Yeah, definitely. I think you make a really good point. It's a really, really good point. You know, as I said, this is kind of more aimed towards the kind of university thing rather than the commercial sector. And I think this kind of forum kind of really grew out of the Insects for Food and Feed conference, which I think
yeah, I was there.
Yeah, exactly. So Insects for Food and Feed conference, who invited kind of Megan to give a talk, and it kind of led to this this happening. But yeah, I think you make a really good point. Thank you for that. Any other questions? No? Any questions online? I don't want to steal over my time.
Just a nice comment.
Oh, thanks, Clive.
It's true, Clive.
Alrighty. Okay. Well, in that case, I think we're on to our next talk. Who's up next? Oh, excellent. Gemma. It's Gemma next. Brilliant. Right. I'll hand over my clicker. But yeah, thank you very much for the questions. Yeah, just that one. Make sure you don't hit that one, otherwise because that's gonna close. Yeah, basically.
Okay, good afternoon everyone. My name's Gemma. I am the research coordinator at Colchester Zoo. I apologise, I'm not an entomologist, as per my welcome slide. I am an amphibian behaviour and welfare research specialist. We unfortunately don't have the inverts at Colchester Zoo to encourage too much invert welfare research at this stage. But what I'd like to talk about today is the changes I've made to our research programme to encourage lower vertebrate research. As an amphibian researcher and a reptile and amphibian ex-keeper myself as well. And hopefully this can give a framework to other zoos and other organisations to help hopefully encourage that research in lower vertebrates and inverts as well. Of course, Eleanor's talked about the university side of things, but I think as those organisations that hold those animals, it's important that we make those steps as well to try and encourage that research from our end as well. So I'll give you what I've done with my frogs basically, and hopefully it will give you some lessons for what we can do in the future with invertebrates. So my undergraduate degree dissertation looked at issues that zoos are facing in terms of holding threatened amphibian species. And as I took over research at Colchester Zoo in September, I've been thinking about those issues and those challenges that I raised in that dissertation. And I've been thinking about how those might be similar to the issues that we face encouraging research in this area in zoos. So I've come up with 3 issues that I think we face as zoological institutions, and hopefully then I can introduce some of the solutions that I've put into place at Colchester and give you some of the stats that show how some of those changes have been taken. So the first issue I would identify is, as Eleanor talked about, a lack of interest from students in these particular species, in lower vertebrates and invertebrates in particular. Gaps in the literature, so those foundations, those scientific foundations often don't exist. Sometimes supervisors and students feel it's difficult to springboard then from existing literature to develop the ideas that they want to use in their research. And finally, there's definitely limited expertise. Obviously, you guys are the experts in the invertebrate world, but from my experience in lower vertebrate research in zoos, we definitely have a lack of expertise in that area as well. So to cover some of those issues and some of those solutions, we'll start off with lack of interest. So there's a lot of work being done in those universities, and I have seen an increase in staff driving those lower vertebrate and invertebrate projects. But there's definitely stuff that we can do as a zoo to try and encourage and spark that interest as well. So we might be producing specialised research priority lists. We might offer and advertise the publication and presentation opportunities to students in the future. Bars or awards are awards that are offered by the British and Irish Association of Zoos and Aquaria, and I'll talk about how those are a little bit more in the favour of lower vertebrates and invertebrates. Fantastically. And there's a huge application to conservation. This, I think, is the most important thing, but I'm going to go into these in a little bit more detail. So at Colchester Zoo, and most other zoos will do this as well, we've produced a research priority list. And these are created by myself, our curators, and our head keepers on different sections. And we look at what issues we're facing on section and how we can solve those with research questions. I run the postgraduate priority list in particular, but I do offer some of these projects to undergraduate students from universities like Writtle that we work very closely with as well. And I've definitely increased the number of projects in lower vertebrates on this list. So they cover an amphibian project that actually extends my master's research, a couple of Komodo dragon projects looking at changes in behaviour in different seasons, which hopefully can give us some insight into how our Komodo dragons use both their artificial and natural UV within their enclosure, looking at growth rates in our young Komodo dragons at this stage, and looking at UV provision for snakes. And before I started this role in September, these were all mammal projects. So even just having these 4 lower vertebrate projects is definitely a step change. I like to advertise, I'm a product of this myself, the opportunity to publish and present your research as a lower vertebrate and invertebrate researcher. I definitely feel there's bigger opportunities there because it's such a new and exciting research. It's got such fantastic applications. I encourage all of my students, but particularly those lower vertebrate and invertebrate students, to look into publication. There are some fantastic zoo-based research journals now that are increasing the output of lower vertebrate and invertebrate research. JZAR, Journal of Zoological Botanical Gardens, is a new one that I choose to publish myself. I'm a big fan of the way that journal works, and Zoo Biology. But there are definitely some more specific journals for lower vertebrate and invertebrate research, loads and loads for herps. When it comes to zoos, as I say, I'm not an entomologist myself, so you might have some other ideas on this, but from what I could find, invertebrate biology is where any zoo-based research seems to be published. I would definitely highlight these journals as a priority because they give you that access to the wider zoo community, so you can reach that wider audience. And luckily, these journals are definitely noticing that gap in the research because JZVG in 2022 released a special issue on fundamental knowledge on forgotten species, for example. And this is actually where I published my first paper. And it was a really nice way to highlight those forgotten species. It included lots of amphib research. There were a couple of mammals that sneaked their way in there, but I suppose they have their own justifications. But it's a really nice way to highlight those species to the wider zoo audience. And if someone was a mammal specialist, they'd definitely still be able to have access to our herp literature there, for example. There's also lots of opportunity for presentation in the zoo industry. I sit on the BARSA research committee, and I'm constantly nagging my students to submit abstracts for our research conference that we hold every year. That's a really nice networking opportunity to meet researchers and other scientists in the zoo industry. But we also have the ABWAC symposium every year, the Association of British and Irish Wild Animal Keepers. That's a nice way for students to access keepers themselves as well, so that they can communicate and share their findings with the people who are actioning those applications directly. So both I would recommend to students to attend because they are definitely a different group of people, but both important to access and to have those conversations with. There are some more specific LBI meetings. The British Herp Society holds a fantastic conference every year. Really great place to present. Fantastic researchers there. Lots of fantastic conversations come out of that particular conference. But BIASA, the British and Irish Association of Zoos and Aquaria, does have specific working groups that might be relevant for lower vertebrate and invertebrate welfare research. So native species working groups, reptile and amphib working group, and the terrestrial invert working group, as well as ABWAC, who hold specific workshops and symposia for different taxa as well. So again, another way to access those keepers who might be able to action those changes on the spot. BASA then yearly offer an award scheme, so you can apply for an award within a different category, and you'll get given either a bronze, a silver or gold award, and there's lots and lots of publicity around those awards. I'm still waiting to hear how a couple of mine did this year, but we've got awards. If it's a bronze, I don't care, I don't want it, but hopefully they all come out to gold. But historically, lower vertebrate and invertebrate projects have done really, really well in all of these categories, and I think that's probably to do with the novelty of those particular project. So London Zoo has done really, really well with their Research Husbandry and Welfare Awards in the past. I'm waiting on my goal for my London Zoo research project, hopefully. Chester Zoo and also London Zoo has done really well with field conservation awards in lower vertebrates and invertebrates as well. Conference presentation awards do tend to highlight these areas as well. This was my certificate when I first presented at the BASA Research Conference. It was held at London Zoo where I did my research, so I took the certificate to see my frogs. They were the stars of the show, so they got their certificate. But there are also photography awards, which have specific categories that highlight the weird and wonderful, and those, of course, do tend to highlight lower vertebrate and invertebrates. I'd also like to highlight a project which was now a few years ago, but a gold award that went to Reese College for their work on the fen raft spider field conservation as well. So a really nice way to highlight a small organisation there who isn't always such a big name in those awards sometimes, but definitely that work made a nice big stamp there and a big print on the community. And I think most importantly, it's really nice to highlight the applications that these projects can have to conservation. Given the conservation status of many of our lower vertebrates and invertebrates, I like to say that any project within this area can have significant effects on conservation, be that a direct application if we're working on application to the field, or work that in welfare and behaviour that improves the care of animals in captivity that might be involved in conservation breeding and potentially translocations as well. So if we highlight those specific groups on the IUCN Red List compared to those mammals and birds and plants, like they were on there as well, but we can see that there is definitely a call for conservation research in those particular areas where zoos can be involved. So we could encourage research into conservation breeding, conservation education and engagement, translocations and reintroductions. And particularly for these lower vertebrate and invertebrate species, where things like translocations and reintroductions and captive breeding are sometimes much easier and much more accessible, that research should definitely be a priority. So our next issue is gaps in the literature. You guys will all be aware you have this in your own field. I certainly remember coming to do my undergraduate project and my NRES project and going, don't know where to start because I want to do some frog stuff, but there's nowhere to kind of get my ideas from. So I like to say that to fill these gaps effectively, to lay the foundation for future research as well, we need to go back to basics. We need to lay that foundation so those ideas can have somewhere to springboard off of that is scientifically robust and that we can trust as well. I did this with my beautiful frogs, and I'm going to introduce you to them a little bit more. This is the Lake Oku clawed frog. It is a threatened species, a critically endangered species endemic to Lake Oku in the Cameroon. It was a fantastic kind of studied species because it's a close relative of the well-studied West African clawed frog used in medical research. Lots of kind of research coming out into their behaviour and welfare now. So given that this was a close relative, it seemed a good place to start. They are only held, however, at ZSL London Zoo. So the work that these guys do to breed and hopefully in the future reintroduce these guys is really, really important. But research on them is currently limited, and best practice that does exist. Well, we just brought out the new guidelines last week before I wrote this, after I wrote this, sorry. But until that point, all of the best practice guidelines there were based off of observation and anecdotes rather than this kind of scientifically robust framework. So I started right at the beginning. I spent a third of my project doing what sounds sometimes completely pointless, writing an ethogram. And straight out of this ethogram, I came up with loads and loads of conclusions that told me so much about this species, as well as this gap in the literature. So by identifying the behaviours of this species, I suggested, and I could conclude, that they had a very, very different behavioural repertoire to that close relative. Now, in the past, we'd look at the ethograms of a close relative. We base a lot of our care, and our husbandry on what we know about that close relative. So having this ethogram alone told me straight away we need that species-specific welfare and behaviour research. We can't be making assumptions based on those closely related species, and we could be causing quite significant compromises to welfare if we do so. I then created, again, sounds very basic, but an activity budget for the species, looking at differences between males and females, frogs in different conditions, different tanks throughout the day as well, and again found big differences between those other species, those closely related species. But using this activity budget, I then, this is my favourite graph on the planet, I then submitted frogs to a routine health check. They were biannually health checked and swabbed for chytrid fungus, and we used that as a stressor event and observed their behaviour after that event, and found in particular significant increases in swimming behaviour. Now, the plan at the time was to compare this with corticosterone levels with the stress hormone for amphibians, but COVID said no. So at the moment, that's still in progress, but keepers have been using this behaviour as a welfare assessment tool, and as a result of this work, have also reduced the number of those routine health checks to just yearly rather than biannually. But having this foundation, at the time it felt a little pointless, but having this foundation, we can then see the applications of that, because I recently supervised a project at the Royal Veterinary College, which used that behaviour that I'd identified to assess the frog's response to changes in their enclosure. And this student changed the background colour on the tank, because there was research in that close relative species that tank background has an influence on their behaviour. But she looked at white, black, green and grey backgrounds and found that there was a stress response to all four backgrounds, but it was less obvious, less significant in the green and grey backgrounds. Another interesting point was that frogs habituated to that background colour over 5 days. So even where there is a stress response, they're recovering very, very easily. Again, this has been applied to husbandry of the species at London Zoo, but really it's important here to highlight that without that foundation, we wouldn't have been able to assess those husbandry changes. And there's now a programme of projects to continue to look into the effect of handling, the effect of different husbandry practices on the species using that indicator. This has been done in a couple of other lower vertebrate species, and you might notice all from the same organisation. So there was a study looking at the effect of disturbance in mountain chicken frogs, again a big conservation concern. Congo Sicilians, they looked at the effect of disturbance and found that prey intake and reduced prey intake was potentially an indicator of welfare, again, that can be used to monitor welfare non-invasively. The same team moved their giant Galapagos tortoises from a quite large enclosure to a lovely new enclosure. If you are ever by London Zoo, do check that out, as it's growing, it's looking fantastic. And they found that, again, using that basic measure of activity budget, they could tell a lot about the animal's welfare, because the activity budgets became more similar to wild individuals in that new enclosure. And their hierarchy was inverted as well to a more stable hierarchy. Now, that, of course, was on my reptile and amphib examples. London Zoo has also put out some fantastic research recently looking at the effect of handling on the Mexican red-kneed spider. And this is the first paper of its kind, really, in zoos. So a really kind of statement piece for us to have this output for. So looking at the effect of handling by looking at behaviour after handling and on control days to 24 hours. Charlotte found that spiders spent significantly more time under cover and had more limb interactions after handling. She did infer that it's not possible to infer welfare implications at this stage, and that more monitoring is required, but it's a really nice example that to say that there is behavioural change in this species based on this disturbance and this handling. So that brings us on to that limited expertise, because you might notice all of those studies came from the same team at ZSL London Zoo. That's not to say there are not teams putting out this behaviour and welfare research in lower vertebrates and invertebrates, but to be putting out that research so regularly, this is the only team that is currently doing that. I, of course, conducted my research at ZSL London Zoo. I learned from the great Chris Michaels, who's no longer in the zoo industry anymore, unfortunately, but really helped me to have that experience of what a good supervisor should be like, how I can be supported by that zoo, and I hope to kind of instill that in how I work with my students in the future as well. So I've got that expertise from him, hopefully, but to try and tackle that issue of limited expertise, it's important that we continue to share knowledge between zoos as regularly as possible. We can do that through collaborative projects, student research projects as well, and committee memberships are really important there. And one thing I'll talk about later, which is my priority at the moment in our research programme, now I've kind of tackled this bombshell, is to look at capacity building for keepers in research as well. So in the past, collaborative research projects have been really successful. It's allowed a kind of standardisation of outputs in these particular areas. ZSL, Chester Zoo, Durham and Bristol worked on all of the conservation breeding work with the mountain chicken frog, and all of that research output was standardised across all of those organisations. At the time I wrote this, we were hoping to do a collaborative research project with ZSL and Chester Zoo, looking at growth rates of our young Komodo dragons, using comparisons in particular with ZSL's humongous Komodo dragon, who has now unfortunately passed away. So they're just settling in their new female at the moment, so the project is on hold, but we hope that in the future we can work with all of these organisations that have these same species to make more collaborative research projects. And we can do this successfully through memberships, committee memberships under BIASA, EASA and WASA. So for those who don't know from the zoo industry, BIASA is the British and Irish Association of Zoos and Aquaria, EASA is the European Association, and WASA is the World Association. And these organisations have tons and tons of different committees and working groups and subgroups that cover a range of different areas, from welfare to education to research to membership to veterinary to native species. Covers loads and loads of stuff. You can be a member of a WASA group and an EASA group as well, which is a nice mix of experiences. So we're going to look at BIASA in particular, and there's lots of different groups on here that might be relevant to lower vertebrate and invertebrate welfare research, but in particular I'm going to focus on the BIASA Research Committee. As a member myself, I can give you a bit of an insight into how we can communicate in the committee to increase those projects. So if we look at membership of the BIASA Research Committee, it's made up of research professionals, zookeepers, curators, educators, academics, and these cover areas and specialisms including herps, mammals, birds and aquariums. At the moment on the research committee, we don't currently have an invert specialist. But of course, if we do ever get calls for research projects looking at inverts, we can definitely communicate with other groups to get support on that. We also have a BIASA research handbook, which does try to highlight the importance of studying those neglected taxa, and lots of research has come out from members of the committee as well, looking at taxonomic bias to support that. And there's tons and tons in that handbook just focusing on aquarium-based research as well. All available online. It's a bit of a chunky document, but it's a nice kind of step-by-step, which I like to refer students to as well. The BIASA Research Committee also works with working groups to create BIASA research priorities. It's not the user-friendliest document, but definitely the terrestrial working group have put a lot more work than some other groups into creating a really useful document here with fantastic project suggestions, as well as experts to contact if you're interested in those fields as well. So hopefully that does support students who are interested in those projects. As a BIASA Research Committee, we look at different applications for research support. Often this is when students want to do multi-institutional research projects. And we had 18 applications for support in 2022, 61% of which were endorsed, but only one was a non-mammal taxon project. I believe it was a bird project as well. So it's still definitely a higher vertebrate focus there. And as a result of that, the research committee urged applicants to focus on those understudied taxa and those who have little attention of and are really in need of that research. We haven't got the stats as to how that's looking yet, but I haven't yet seen a low vertebrate or invertebrate application come through. And then to focus on capacity building could help with some of these issues. Our zookeepers are the experts in the taxa that they work with, and if we can give them the tools for research, then we can definitely enhance our research output. We are working with our keepers on trying to develop those research priorities more significantly, rather than asking for how does an animal use an enclosure, how is it affected by visitors. We want keepers to come up with problems that can be solved by research as well. So I'm designing a whole training program specifically for that to help keepers realise the potential of research, and hopefully that will improve the suggestions that we come out with. But we also want them to be conducting research themselves. Keeper research is a really, really valuable tool. Keepers know those animals better than we do. They can tell those individuals apart. Animals know keepers as well, so we reduce that observer effect. And also they are in a position to manipulate conditions if necessary for that research. But we want them to be sharing findings themselves, positives and negatives, really important to share with the zoo community what went well, but also what went wrong and what not to do yourself next time. And we want them to be engaging really well with student research findings, which hopefully they did at our event last week. We had keepers come down and listen to research projects from students over the last year, ask lots and lots of questions. It was fantastic, but we want to make sure they're continuing to engage in that research, which of course is for their benefit, for their animals' benefit. And then when they've been able to engage in the research findings, then we hope they can apply those research conclusions to the way they care for their animals. So after implementing all of this at Colchester Zoo, some of it's still in the works, but definitely at the first few stages of that. Did it work at Colchester Zoo? Well, preliminary findings suggest that we have had an increase in lower vertebrate projects. So between the years 2021 and 2022, we had 31 projects conducted at Colchester Zoo, none of which were conducted on lower vertebrates, all mammals basically. But upcoming projects this year include giant tortoise aggression and enrichment. Serena, who helped organise the event with Eleanor, is unfortunately not here, I'm afraid, because she's at the zoo watching her tortoises right now. But a really fantastic project there, which is really, really valuable for our keepers to have those conclusions of. Anglia Ruskin are currently looking into seasonal changes in Komodo dragon behaviour from the priority list. That will give us a bit of an insight into how our Komodo dragons use artificial UV during the winter, but also when the roof opens in the summer, how are they using that natural UV as well? Where UV is a really exciting area of reptile research at the moment. Hopefully we've got some good insights there. And I've also offered to the University of Suffolk the project looking at amphibian behaviour and indicators of welfare. So an extension of my master's project looking at our dark frost species at the zoo. All of the references are on there. They're really fantastic papers, so definitely go and have a look. Even just, obviously, you might want to look at the spider one in particular. But all of these references are really great examples of framework and foundation research that hopefully can be used in the future to kind of investigate those effects further. But once we've got those nice ecograms and activity budgets, it definitely provides a nice scientific and robust foundation for research in the future. But that brings me to the end of my presentation, so thank you very much for listening.
Thank you very much for a brilliant talk. Again, another talk that I'm sure some of you wouldn't have expected to have at an entomological conference, but really, really glad you could be here. Like, it's really interesting to see the kind of zoo priorities, and also it's great to hear that you're kind of interested in supporting research into these areas. Does anybody got any questions? If not, I've got one to kick off with. So, you've talked about these brilliant kind of resources you have at the zoo, opportunities to do these really important experiments, a lot of species that most people don't have access to. For entomologists and welfare researchers here or online, if they wanted to, collaborate in some way, how would you recommend getting into
Absolutely, yeah. So we're really keen. Part of extending and expanding the research program is to be more involved in research and to increase the calibre of research that we're involved in as well. So historically we used to tick a box, send an application form off, let the students in, but it's really important to me that we've got lots of supervision support there. As Eleanor said, really, really important to have, I think, from both sides in a student research project, but also to have research projects coming from other areas that are not just student research projects. So if you have an application, Andrew already talked about potentially looking into olfactory profiles of different reptiles, for example. I didn't put my email address on there, but my email address is gemma.dyers@colchesterzoo.org. I can write that down for anyone who's interested. But a fantastic resource there that I'm keen to open up to anyone who can find a use for it, really. If it doesn't feel like it's got a zoo kind of application directly, you know, it's got a conservation application and a medical application, it's still valuable for us, and it highlights the work that zoos do outside of just exhibiting animals as well, and how we are becoming centres for science and conservation. So absolutely, any questions or requests for research, please do get in touch, and we can have a look about how that might work in Colchester Zoo.
Any other questions on the floor? On silence, entomologists are
What's
going on? Got a couple of questions online. That's great. So I'll just read it out so people who can't read the writing on the back. General question, perhaps for closing session. Being online and not being able to see the audience, but for those who've asked questions, sounded young. Except I think I heard James. Speakers without exception were young. Do you sense that it could be an uphill struggle convincing the older die-in-the-wool entomologists for the importance of insect welfare or ethics getting involved in stuff? I don't think this is necessarily aimed at you, Gemma.
No, I would say though, however, from a hurt point of view, 100%, I found older herpetologists a little more resistant to welfare research. Very sad in that way.
Maybe I can open this one up to the floor. Does anybody from the floor have any comments on this? Anybody? Megan?
Yeah. I mean, definitely same for me, that the conversations that I've had with younger entomologists, they tend to be interested, they want to change their practice in many cases, they feel really motivated, they've already been thinking about these concerns and are just glad someone's discussing it with them. When we held our welfare session at the ESA last year, we had about 90 people in the room, and it was very young, the crowd. So I think like my personal experiences and that anecdote suggests that it is young. I do think one thing to be careful of is with many of these older entomologists, they have entire careers behind them of doing particular kinds of work, and you might feel very personally attacked by this conversation to some extent, that you're being judged. And I think the more we can do as a community to not imply that there's judgment in this conversation, but rather that we as a community are just looking to improve our practices without targeting any particular community or group of researchers for what they do. I think the more we can do that, the more likely we are to get those folks also on board. And there are some older entomologists on the board of the Insect Welfare Research Society, as well as some of my committee members actually during my dissertation, who I think have demonstrated they're pretty open to this conversation as well. So I think a lot of it is how we choose to lead the conversation on welfare and ethics with those folks.
Yeah, very well put. We have two more. I think you were first, and then
I'll make a comment. I think got to be quite careful how you interpret older entomologists, because I think they have a very different background. In that I think many of them are originally naturalists and probably have come from a very different place. And I think for some of those older entomologists, I would say they probably have a greater appreciation for that natural biology aspect of invertebrates that we perhaps, in this younger generation of invertebrate devastation, perhaps don't have. I mean, many of those older entomologists grew up as bug keepers and people collecting bugs as children, and I don't think that it certainly does still happen, but it's not as common. So I would say, in my experience, of course it's a mixed bag. You have some, I've known entomologists in their 90s, known entomologists in their 10s. I think some of those entomologists in their 90s have a different appreciation. So I wouldn't necessarily say, invertebrate welfare is a young person's game. So the Invertebrate Society, for instance, is 25 years old. It was started by a lady called Mary Branker. I think there's actually some appreciation for welfare of invertebrates is quite old, in truth. These questions have been asked many, many times over decades and decades and decades. So I think it would be unfair to just say it's a young person's game only. I think actually it's quite mixed in my experience. But from my anecdotal experience, I've seen very stuck in their ways entomologists who don't think invertebrates feel pain, but I've also seen the opposite as well. So I think it's quite mixed, to be truthful.
Yeah, just to follow up what you're saying, I've been very impressed by some of the lovely emails I've received by a whole range of different academics at different stages, a lot of whom have been doing, really great best practice things for a really, really long time. And so, lots of wonderful people doing lots of wonderful things. I think there's one more you want to add anything?
Yeah, we're in black soldier fly, and most of my customers are farmers. And as we know, the average age of a farmer is 63. So I just don't think it's real. And most of those are really interested in keeping their animals, whether it's cows, pigs, chickens, or black soldier fly. And they're really interested in the welfare. So I think it just depends who you're talking to. I think that the researchers here tend to be younger and so talk to younger people, whereas I don't. I talk to, don't get me wrong, there are some very young ones as well, but there are definitely older people who are interested in insect welfare.
Last comment on that would be that I think it's also sub-discipline specific. Like, you're right, the veterinary folks have been thinking about this for ages. You can find tons of literature going back 20, 25 years. The health people, like the disease people, have been thinking about this for ages. People who focus on diseases insects get for the sake of insects, not people focusing on like vector biology. But when you get into, and like ethologists, most of the older entomologists that are on our board are people coming from a behavioral or a natural history background. When you move into physiology and neuroscience, I think you start to get a slightly different trend. And this is classic when we look at other groups of animals as welfare extensions have been made. It is often the ethologists at the forefront and the physiologists and neuroscientists lagging a little bit behind, depending on myself. So I think like there's some of that at play as well, alongside potentially the age thing. You have these disciplinary distinctions that might kind of co-bound the age question.
I would make the comment that the Royal Entomological Society's special interest group in veterinary parasitology is more about killing them, which I always found I found that very entertaining when I first joined the Veterinary Entomology Society, because obviously my perspective was completely different. So
it's the same in the ESA section move, which is our medical and urban veterinary.
So yeah, great comments. Thank you very much for sharing your thoughts, guys. And thank you for that very, very good question, Richard. It's had some good conversation, which I hope kind of translated well over audio as well. So we've got a question now from Lizzie. I think this is back to you, Gemma. Thank you for a great talk. The language that we use shapes the way we think. Lower vertebrates suggest that they are lesser in some way, so maybe some less exciting to students. Do you think that we could talk about these animals without using value-laden terms such as lower? Could this be
absolutely. Yeah, I use the term lower vertebrate and invertebrate in this talk in particular because in a zoo world, that's how we tend to group keepers who work in that particular field. For example, I myself would historically call myself a herpetologist. So I would normally use the phrase herptile, but I didn't know if that was a commonly known term. That's why I think there is a move also to reptiles and amphibians rather than herptiles as well. And also to highlight the separation between the two groups as well. We shouldn't always lump them together, and people can have specialisms in either. So there's definitely a shift there. As for thinking about reptiles, amphibians, fish, and invertebrates, currently they are still termed lower vertebrates and invertebrates. But I definitely agree we should be looking for a change in how we group them, because more and more research is finding they're not necessarily lower in any way than mammals and birds.
Great. All right. Well, thank you very much.
Sorry, one more question. On the topic of that, because you do see it all the time whenever there's like keeper vacancy or whatever, LVI. Do you think that tagging as sort of almost like just something attached to the end of it, the invertebrates onto it, seriously dilutes the knowledge of invertebrate keeping within the zoo, and maybe there should be a push away from that?
Definitely.
Because you look at a job vacancy for LVI, I've looked at them before, so I love it. I've always thought, one day maybe I'm going to go down that road. But it's just literally like 2 bullet points at the bottom of a job description. Everything else is what you require in terms of looking after specific area within amphibians or reptiles, and it's like, can you also feed cockroaches at the bottom sort of thing. I think it does seriously dilute the knowledge zoos have got.
I think it's still a historical thing of a lot of invertebrate keeping in zoos historically was looking after your reptiles and amphibs and your feeder insects as well.
Sure.
There is still, as far as I know off the top of my head, there is only one team that I can think of, which is a purely invertebrate-based team, and that's at London Zoo. at the moment. I think there's also currently not, collections don't have a big enough invertebrate collection to have a whole team run that section, which is why they get lumped together. I mean, I'd like to see as an amphibian keeper, and I definitely have a preference for amphibians and reptiles. I would like to see a separation of reptiles and amphibians. So I would like to see aquarists be separated from herpetologists, and herpetologists be separated, and invert specialists be separated from that. But I think we still have a lot of way to go in terms of collection planning to have big enough invert collections, to have to have the interesting exhibits as well that warrant them, because we don't just want to pop them into a collection for the sake of it. We need to have the helpful interp and the educational journey to go with it to help people find the interest in invert. So if we're going to do it, I think we need to do it properly as well. So it might take some time, but there's a lot of kind of big heads out there that are thinking about these ideas that I think as we're starting to think more holistically, as an educator, I'm thinking about exhibit design all the time and how we can be there at the beginning of the journey. So hopefully these exhibits will start to expand. And even, Chester Zoo has a lower vertebrate and invertebrate team, but they have some incredible invertebrate exhibits, and the way they mix them up with, with mammals, you stumble upon these exhibits. That's why we at Colchester have never had a reptile house, because people don't like a reptile house. They revert to a building full of snakes, but you stumble upon our reptiles around the zoo. So hopefully that clever exhibit design will warrant a bigger invert collection across zoos, and that will warrant no separation of teams. I'd like to see it. I reckon probably not.
It's my twilight years,
Yeah, absolutely.
Thank you.
Thank you.
We're running quite a bit of time as well. So just a time for final tea break. So do grab another cup of tea, and then we've got our last session. After which, so I know some speakers are dashing off, so do get people's contact details and that kind of thing. And then after that, we have got a pub for anybody who would like to go to the pub. Also, people online, if you are within driving distance of a pub, you are very welcome to join us in the pub afterwards. But so go grab a cup of tea, and then we've got our last two speakers of the day. Thank you very much for your chat. Again, for those of you who are online, it's the black soldier flies again. But so we're now back, and we've now got our two last speakers of today. So first of all, we've got Benjamin back to talk about a different area of work, and then followed by Bob as well. So Benjamin, go for it.
Okay, fantastic. Start. So hello everybody, I'm just going to give a very quick talk on invertebrate medicine and the sort of welfare considerations I have. So I'm going to talk about a little bit about who I am and who this strange bearded man is squawking at you. I'm going to talk a bit about invertebrate medicine, and I'm going to talk a bit about invertebrate welfare. Now obviously, 10 years of my life, 98% of all species. This will be very quick and introductory. If you guys have any other questions and you want to go into more detail, I can talk to you for several hours later on. So this is who I am. So I have an academic background before my veterinary degree. So a few degrees for my sins before my veterinary degree. I'm actually a small animal and exotic vet. I primarily work as a locum, about 3 to 4 days a week. And then 1 day a week, I undertake private research and pursue clinical work in invertebrates specifically. So I'm a member of the steering committee of the Veterinary Invertebrate Society and a council member of the British Veterinary Zoological Society. So I'm involved with quite a few different academic societies. I'm a member of a lot more, so just skipped ahead. So why treat invertebrates? Well, I think, there's increasingly more people keeping these animals as pets, both within private and commercial collections. There's a huge diversity of the species being kept in captivity. So tarantulas, scorpions, phasmids, everything. And for instance, in tarantulas, a good 200, 250 species are kept in captivity. So that's about a quarter of all the species reported. So actually, there's a great deal of these species being kept. Beyond that, that it's really underappreciated how many owners there are. And as vets, we have an obligation to provide those owners with the ability to keep their animals well. So I do love this quote here. I know, so Haldane is an evolutionary biologist, and he said God was inordinately fond of beetles. So the sheer number of species beggars belief. There are more jumping spiders than there are mammals. There are, if you were to take all species of beetles, there's 8 or 9 times more than there are vertebrates. So huge number of species. So,
and this is a
forgive that. So this is an image of the British Tarantula Society exhibition. To really put it in context, there are really an unappreciated number of owners. So that's more than 3,000 or 4,000 people undertaking, buying and selling insects and tarantulas and other invertebrates, crabs and snails. So there's a very underappreciated group of people owning these animals that it's not necessarily acknowledged that there is the aspect of this as well. So why is veterinary input needed? So I think number one, so this is some unpublished data. It's in the works, a survey of tarantula owners, of which we had 1,060 responses. We actually found that about 85% wanted more invertebrate, wanted more veterinary input. So these are, as we all know, many of you academic scientists are very important as animal models within science, and they're important commercially, industrially, and as educational tools. They're important as pollinators, as a source of food, and they do play a very significant conservation role. So invertebrates are going extinct, between 10 and 16 times more readily than vertebrates, actually. They are actually a very key important part of habitats and habitat preservation. So there's no point saving the elephant if you don't save the dung beetle, because all you're going to have is an elephant you reintroduce surrounded by crap. So we have to consider the whole picture. And actually, invertebrates represent one of those few instances where a disease has wiped out a population, and that was the primary reason. A case of microsporidium in partula snails. So actually, health in these animals is more important than we all appreciate. So one of the very few extinctions due to disease. So what is invertebrate medicine? Now, this is one of these slides I put in my presentations for students, because really it's very, very simple. I think we make this much more complicated. That's what invertebrate medicine is. You have a tarantula, and there's this adage which is pretend it's a cat with extra legs. I've done that to vets and to vet students, never, because actually the truth of it is they are animals. And though the mechanisms and the functions, or the mechanisms and the biology might be different, the function is often the same, actually. So, what are my diagnostic tools? So it's a cat. Really, I do a clinical examination. I use the same diagnostic tools we would use in anything else. So hematology, biochemistry, histology, culture sensitivity, so I swab, and I use imaging, so radiography, ultrasonography, MRIs, that sort of thing. It tends to be more than your clinical exam. So let's have a look at an example of that. So on the right here, we have a pseudo-intermediate scorpion, and on the left, we have a jumping spider. So if you can see here, if we get the laser sight up, sorry about the people online, you can see the eyes, you can see the mouth parts, you can see these, you can actually see the heartbeat if you look very closely. You can see the hepatopancreas diverticulum on both sides, these two brown organs. Here you can see these large eyes and large jaws. This is a male jumping spider because it has its balls on its face. So, and to put it in context, that's how big these animals are. So they are small. So what I use is I adapt my traditional approach. These animals are incredibly small. Actually, if you think about the size of this scorpion, I am probably a good 500 times bigger. So it then represents one of the challenges of actually trying to treat and manage these animals. Again, this is me taking a blood sample from a tarantula. So I apologise for the wobbliness because this was when I was still a student. But it just illustrates that you can take a blood sample And this same site can be used to give fluids as well in an emergency situation to a spider. And I'm taking quite a lot actually. And then this is what you get. So if you take that hemolymph and you put it on a slide, you'll see this is crystallization, protein crystallization. So because their blood is primarily hemocyanin, rather, so that their oxygen carrying capacity, oh, I keep skipping ahead, isn't actually in cells, it's actually free in the plasma. So it forms crystals. But you can also see other features on here. You can see these little dots, which are little bits of bacteria, which is a variation of normal in many tarantula species. And again, this is histology. So histology is the process of taking tissue, fixing it, and then making a series of slides, and then staining the slides. So this is a stick insect. And you can see lots of different anatomical features. So these are Malpighian tubules, which are the equivalent of kidneys. We've got here, you've got neuroganglion, which we've all been discussing earlier. This is an eye of a phasmid, a compound eye. On the upper right, we've got the stomach of a phasmid, and you can see these large air-filled organs, which are the respiratory tracheae. And actually, you can use this tool to see pathology. And again, this is on the left, we have a tarantula of a mass, or what appears to be a mass in its opistosphoma. And on the right, we have a normal tarantula. So you can see the difference here. So this ended up being some retained cuticle that then formed a sort of granuloma around that retained cuticle. So pitfalls in the solution. So these are small animals. And I think that's one of the reasons that sometimes it can be more difficult to assess welfare, because we're used to assessing welfare in comparatively much larger animals with a different biology. And again, our other issue is, certainly from a veterinary point of view, what do we know what we're looking at is normal, or if it's pathology? And actually, we don't necessarily have the data to say that. And many of our clinical tools are validated for mammals, primarily humans. That even applies to birds and reptiles. If we take a swab of a reptile, we're growing it up primarily in a lab in conditions for human flora. So we have the same issues with invertebrates. So a lot of what I do is I take several tests to validate that what I'm looking at is genuine pathology. And it's one of the big challenges of invertebrates from a diagnostic veterinary point of view. But then we also have lots of unique features which they simply do not mechanistically have the same mechanisms that a mammal or a reptile, or even a fish, even quite a different vertebrate has. So they don't have albumin, they don't have many of the same biological pathways. So really they have an exoskeleton. So really it's looking at those things and looking for that parallel function, and then using that and pathology and what I see in front of me to ascertain what is pathology, and then try and treat it with the same tools that I have for cats and dogs, and trying to apply that to invertebrates. So, and this is just an example of treatment being successful. So this is a tarantula with a quite heavy parasite burden. The video of it. And this is, this particular tarantula is something called a warm Murphy, it'll bite your face off. So it required anaesthesia in order to actually remove the mites, but once they were removed with an alcohol wash, successful treatment, returned to the owner, very happy with it. So this brings us to invertebrate welfare, and there's this term. Now, I think this is where we're going to get a little bit philosophical, because, by what measure is a non-human is actually something I got from a website called TV Tropes. If anyone's heard of this, take great care, it will devour dozens of hours of your life. But really, this is a website based around tropes in media, so stereotypes, common aspects of stories. And what measures of non-human, what this trope is about, is it's often about, when you're playing a video game, you're killing all the zombies, or all the robots, or all the aliens. And there's never a question about how sentient or how meaningful those aliens are. You're killing all the goblins, not realising all the poor goblin, now orphans being left. I keep returning to this phrase, by what measure is a non-human? Because really, one of the problems with welfare is our understanding of welfare is based on us, on humans. And this becomes a problem even for reptiles and birds, of which we know a comparatively huge amount more. You know, we are debating the forms of analgesia in reptiles, and we've got equivalent biology. But the issue is not necessarily, you know, when we're thinking about invertebrate welfare, are we thinking about vertebrate welfare, and are we trying to apply that to an invertebrate? And that's not an unfair way of viewing it. Because if you were to say, if welfare is only important if it's similar to human sentience, there are many animals that suddenly don't have any welfare issues. So there are wonderful ways of getting around this by just saying, well, welfare is only something if it's mammalian, and then you discounted all species. But I guess our problem is vertebrate suffering what invertebrates experience, or are we talking about, and is that how we're going to assess welfare? So the issue is not actually welfare. The issue is how we measure, how we think about what welfare is. So really, this is a much more philosophical, much deeper question, and I think this is why it's continued to be a humongous challenge for scientists for a very long time, far before our forefathers, and I suspect will be an issue for many decades and hundreds of years to come. So the real question is, what is pain and suffering for an invertebrate, and how can we view it from a vertebrate's perspective? So there are, you know, many reasons why welfare is important. You know, number one, some of them have got genuine legal protection, cephalopods and decapods. So from a legal perspective, we need to consider their welfare. From the ethical perspective, if they can feel suffering, then it is unethical. So which is why it's very, very important to say that, not stating the obvious. And I think from a pure production point of view, if we're looking at doing, again, I'm just repeating what's already been said, but if we're looking at trying to get productivity from these animals, generally speaking, welfare is associated with good production. These things are not mutually exclusive. I think we sometimes like to think of high welfare production as being mutually exclusive. The truth is they're actually exactly the same. There have been in many, many animals, from cattle to sheep, to dogs, to everything. But then there's also this other aspect of things, which was brought up to me by a student a few years ago, which is, does it matter? Does it really matter if they can experience pain and suffering? Because if you have an owner that owns a collection of spiders, and those spiders are dying, it doesn't actually matter if they are experiencing pain and suffering, because the owner is experiencing pain and suffering. So I know it's a very bizarre way of looking at it, but actually this is more complicated. There are more stakeholders in this question. If you've got someone who's on the brink of depression and all of their spiders die, actually we've got another welfare aspect there that we often don't appreciate when we start to think about the companion animal aspect of this question. So how do I assess welfare? Again, I'm probably going to be repeating something many of you know very well. The first argument we often make is an argument of analogy, which is, I think, many papers, particularly Elwood, has made this argument, and I know he's quoting older people before him. But essentially, it's the argument that, now, if this was me, would I have good welfare? Now, there are lots of flaws to that. The same thing applies. If I poke a dog in the nose, that's not going to be great for the dog. So probably, if I poke a tarantula in the nose, that's probably not also going to be something it's going to enjoy. So and then you then have the argument of direct comparison. Now, is there a biologically analogous situation that I'm looking at that would be associated with poor welfare in a situation where we know that that animal or that situation could suffer? And this is where invertebrates are very challenging, because we don't always have those direct comparisons. And then we have what's often referred to as the 5 freedoms. It's now becoming the 5 domains, where we have freedom from hunger, discomfort, pain, injury, disease, express normal behaviour if they're in distress. and it's then trying to apply these domains to invertebrates. So let's go a little bit into an example. This is a mantis. It's in an enclosure. It's currently exhibiting normal behaviour. That's, I would say, able to express normal behaviour. It's not obviously diseased at the moment. So you would see a cat and dog doing this sort of behaviour, to be entirely honest. And let's go to, say, a diseased example. So this is a example of a mantis with a presentation called black eye, which is what it's known by in the hobby. In truth, when you look at these cases of black eye, you can have a combination of different causes: fungus, fungal disease, bacterial disease, and I've got a suspicion of reproductive involvement. So this animal has got inflammatory lesions. If this was a dog, this would be a dog with bulbous sort of bruises all over its face. Has this animal got good welfare? If it was a dog, I feel like it would be suffering. So that would be the way I would be approaching this animal. Right now, this particular animal was eating nicely. It was struggling to move up in places because it was essentially the end of its legs were beginning to lose some of its grip. So in this case, I started introducing essentially ways that you could get around its enclosure without having to go through that process. This is a more extreme example. So actually, if you imagine your eye, you've got a massive lesion this big, and it's beginning to affect your vision. Actually, if you look here, you can see that little white bit is actually a little mite. So has this animal got worse welfare than the previous animal? I would say yes, because the pathology is getting to the point that actually it's beginning to struggle. This particular animal was being given honey water, essentially, and doxycycline. So I'm beginning to try, I've addressed those issues and beginning to try and follow on from that. Again, I've just got this cool image, which is of this particular mantis. You can see the melanisation lesions here. You can also see the respiratory tracheae, the internal organs move, which I just think is cool. So it shows you that if you actually use if you look at these animals clinically and do a clinical exam, it does require a microscope, a stereo microscope, which all your entomologists know well, I suspect. But actually you can start to think of these animals as you would cats and dogs. So whistle stop tour, hopefully I'm on time. There's a lot we don't know about invertebrates. I'm endlessly being taught very harsh lessons about what I don't know. By what measure is a non-human? I think it's something that I always keep returning to. How are we actually thinking about this? What is our measure of what these animals actually are? And I think the actual truth of it is we can diagnose disease, we can see clinical signs, and we can treat these animals and facilitate positive welfare outcomes. So there's a lot of questions I think we have, and I could go on and on and on. I'll just quickly acknowledge, so it's not just me. I am supported by many, 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 many people. So Steve Trim and Stephen Carroll Trim, the team at Benentech have endlessly supported me. Lots of people involved with the Art of Twig, the British Tarantula Society, a whole plethora of different pathologists, Mark Stidworthy, Mary, Debbie, Simon Sapiro, lots of people at the Royal Veterinary College, a whole set of people at the Veterinary Invertebrate Society, including the Coopers, if they're in the audience. Hello, Coopers, and my wife, who's the most patient of all. So, we've got any questions?
Big thank you. That was awesome. I'm so glad that you could come. That was a really, I just never really thought of it from the vet perspective, and that's really, really cool. I'm afraid we're running a little bit short on time. So, yeah, so you managed to escape that, but hopefully you're going to come later and we can grill you all on the questions later. Sorry? Yes, you did, but I didn't have everybody in there because I couldn't read my watch. So, perfect. And I'm really, really glad that you brought up it being a philosophical problem and we need to have a philosopher, which is why we have brought along a philosopher. So I was really glad that Bob could come along and kind of close off today, because I felt like we could really have this discussion without kind of acknowledging the fact that philosophers are super important here. So we're really glad that you are here to chat to us today, Bob. And whenever you're ready, you know, the floor is yours.
Fantastic, folks. All right. So can everybody, oh, that's not what I'm trying to do. Goodness gracious. You would think this is my first time in life doing this job. Okay. Can everybody hear me fine? And can everybody see this presentation?
Yes.
Tremendous. Okay. So first of all, thanks so much for including me. It's great to be here. It would be greater to be there in person, and I apologize that I can't be, but I'm thrilled to be able to participate in this particular way. We're going to talk a little bit, you know, so much we could talk about with that. The last talk, very interesting. I really enjoyed and appreciated it. However, I will have to push us in a different direction, thinking about how we're going to make some of these trade-offs. And here's what's motivating the project that, or the talk that I'm bringing to you today. So across lots of taxonomic groups, right, not just insects, not just invertebrates, even, there have been these questions raised about tensions between our various goals. On the one hand, we care about conservation goals of all sorts of, right, many different kinds, promoting biodiversity, whatever. We also care about individual welfare, or at least the members of this particular group care about individual welfare for insects. And we want to figure out, well, okay, how do we deal with these tensions? How do we deal with the fact that our values pull us in different directions? And ultimately, what that is going to lead us to ask is the following question, like, well, what trade-off should we be willing to accept? When have we done enough for welfare? When have we done enough relative to, say, our goal of promoting some biodiversity goal where we're, you know, collecting insects because we want to understand a particular biodiversity related problem and then identify solutions and monitor progress toward those solutions, et cetera, et cetera, et cetera. But we want to know, like, what trade-offs should we be willing to accept? How do we balance these competing goals? And right now, we do not have that framework. It doesn't exist, right? What we have instead is just the call to pay attention to the welfare of individual insects and not much more. Okay. And so what we're trying to do today, what I'm just sort of gesturing toward, what I want to encourage us to think about is, how do we do better? Like, what does the research program look like that takes us from raising awareness, which is where we are now, to making principled, thoughtful trade-offs, which is where we would ultimately like to get, because we are not going to get to a point where our values are not in tension. And instead, it will always be about balancing and negotiating tensions between our values. Now, one convenient thing is that we do not have to completely reinvent the wheel. Others have thought about this, okay? So what we've got here is something from the World Bank, which is a document that was produced some years ago now to help people think about how to do assessments of the value of conservation initiatives in you know, more traditional environmental contexts, right? Where you've got a particular ecosystem, suppose you've got a forest with a river running through it. And you're trying to figure out, how do we price, right, the significance of a particular conservation initiative? How do we value all the different things that come out of this? Because of course there are lots of values associated with this forest and the river running through it, right? There's the value of the biodiversity. There's the fact that people use that water. There's the fact that people want to go kayaking. There's the fact that you can harvest stuff from the forest, et cetera, et cetera. And what we want to do is take all of these different things that we value and ultimately put them on a single scale so that you can make comparisons. Because otherwise what happens is someone says, well, I value recreation. And someone else says, well, I value logging. And we have no straightforward way of resolving that disagreement. We don't know how to say, well, this is how much of that the one is worth, right? So what the economists tell us to do is convert everything to dollars. Of course, the economists tell us to convert everything to dollars. That is the way they handle all problems. Now, this is not a criticism. This is also not the only option. However, it is the way that traditional cost-benefit analysis proceeds, where we try to price all of the things that we value so that we can end up with a common currency, in this case, literal currency, right? Dollars, that allows us to make comparisons. So we start off with this goal. Goal is identify all the things we value and make hard trade-offs between the various competing goals. We realize, wait, these things don't come in a common unit. We don't know how to compare them. And so what we try to do is convert them to a common unit. And the economists traditionally have done that in financial terms. Now, let's just flag, let's be honest, let's get straightforward, right? Like lots of us are uncomfortable with monetizing the, things like individual welfare. We don't want to think in those terms. And we also don't relate in monetary terms to things like biodiversity or the value of an ecosystem, right? We have a completely different framework mentally for thinking about those kinds of goods. And on one level, we want to respect that and take that very, very seriously. There is something uncomfortable about treating things that do not in any way feel or seem fungible as though they are fungible and can just be expressed in dollar terms, which of course can always be swapped out. But as important as it is to take that reaction seriously, and I take it very seriously, we also want to recognize we are always making trade-offs. We are always making judgment calls about what things to prioritize and their relative worth, even if we don't explicitly do that, or much less explicitly do that in terms of dollars. So we're not avoiding trade-offs or avoiding approaches to trade-offs simply by not wanting to talk in terms of dollars. What we do is we just do it intuitively and in a way that is hard to explain to others, in a way that captures our own values, but may not actually be able to be communicable to a broader public. So the advantage of converting to a common unit, the way the economists are trying to do that, is it gives us a way to talk to others, to communicate clearly how we are thinking about the various values that are competing for our time and attention. Okay, so let's recognize this. Some values are much easier to convert into dollars than others. Some things we care about are much easier to convert into a common unit than others. So if you think about here on the left side of the slide, you're looking at the direct values. Some things actually have a market price, like certain consumption goods. If you're trying to figure out, well, I wanted to log that forest because that timber has a market price, right? Well, now we can actually go and look and see, well, what is that price? And we can use that as a way of figuring out one of the values that is at stake, namely the value of timber, when we're looking at that forestry case, all right? As we move to the right on the slide, we get to things that feel harder to price, right? And that historically have been, in fact, harder to price. So when we start talking about the intrinsic value of things, how much they matter in themselves, independently of how much use you can extract from them, it becomes much more challenging to do the pricing. However, when we're talking about things like welfare, that's often what we actually care about, is that non-use value. Because although it's sometimes true that welfare is, as we say, instrumentally valuable, you can get things that you otherwise care about from focusing on welfare, that's not the only reason we care about it. So instrumental value of welfare are things like we were discussing in the last talk. The fact that it makes an owner happy is an instrumental value of an insect's welfare, or tarantula's welfare, whatever, right? The fact that it might be sometimes valuable for a production facility to use high welfare standards, right, is an instrumental value of welfare. But for many of us who are motivated by welfare concerns, we're not focused on the instrumental value. We care about welfare in itself, right? We don't want to cause harm. We don't want to cause suffering. And if that's our motivation, then there is no other thing that we can point to use to price welfare, to compare it to these other goods that we care about so that we can make decisions when we're trying to promote our various goals, Instead, we're going to have to think about the intrinsic value of this thing and try to assign some kind of number to it if we want to be able to do this larger kind of analysis. Now, there's a lot going on here, and we don't have to think about every aspect of this. But what I want to flag to you is the fact that the way economists have typically handled this is by looking at people's willingness to pay, right? Now, willingness to pay is an imperfect metric for a lot of reasons, and there's a whole bunch of interesting philosophical and cultural discussion to have about that. However, it's important for us to understand is that when we try to figure out, well, how much does something matter to people? One effective way of getting at that is by thinking about what they are willing to spend. And not necessarily just monetarily, but in terms of their time, which itself can be priced, or in terms of other kinds of resources, which themselves can be priced. And so what we do is we look for ways to capture, in monetary terms, the sacrifices, the costs that people are willing to bear as a way of getting at the values of things. And to give you, just to go one step down in this little chart here, two basic ways of doing that are using revealed and stated preferences. When we're using stated preferences, what we do is we ask people, we go to Megan and we say, Megan, tell me how much you care about, cricket welfare. Tell me what you would be willing to pay to avert, say, 1 hour of this following negative welfare state in a cricket, right? In a particular cricket. And we might get a number out of that, right? She might be able to respond, or then we would survey a larger group and we could get their responses and we could aggregate them and we could figure out, well, how important is this value to these individuals? And of course, there's going to be a lot of variation. But you aggregate across and you can get some kind of starting point valuation that you can use for the purpose of public discussion about the trade-offs that we might want to make, okay? Or instead of doing direct surveying, right, asking people their preferences, we can try to assess their revealed preferences, right? What things do people do independently of what they say that reveal how much they are willing to pay for a good? Right? So we can look and say, all right, let's go into the lab and let's find out how much time people are willing to spend to avert possible negative welfare states. So they're willing to say, freeze their bugs to avoid the risk of causing unnecessary suffering before they do some procedure. Right? That's their strategy for anesthetizing the insects. And that takes them an additional, say, however many minutes. And then we can think, okay, well, how much is their time worth? And what were they willing to just do to alleviate this possible harm? And that is the price, right? That is a way of getting at the price. I mean, of course, it's not, I'm using this expression, the price. It's not the price, but it's a proxy. It's a way of getting at the value of that thing so that we can start making comparisons. Okay, so now remember, where are we? We started with this big thought. The big thought is, hey, people are calling on us to be more careful about individual welfare in the context of pursuing other goals, like promoting biodiversity. We don't actually know how to put biodiversity and individual welfare on a common scale so that we can say, here are the sacrifices it's reasonable to make or that you ought to make for the sake of individual welfare in pursuit of some other goal, right? Here are the ways that you should curtail your pursuit of a biodiversity initiative, for instance, for the sake of individual welfare. So what we are after is some way of putting these things on the same scale, strange though that may be. And the economists tell us, okay, well, one way you could do that is by assigning prices, right? Assigning values in monetary terms to the goods in question. And now what we're pointing out is when we think about welfare, many of us value it intrinsically. It's a thing that we don't value for some other reason, we value in itself. And so what we want to try to do is figure out, well, what do you do when you value something intrinsically? How do you assign a number to that? And the strategy that the economists use is to use people's willingness to pay for the intrinsic good in question. And you can get at that in a couple of different ways, by asking people and by looking at what they in fact do, their revealed preferences. And then of course, in their various strategies for doing that. Okay, well, what are we going to get out of this? What are we going to get out of this? What I want to leave you with, my main goal for today, again, we're not actually putting the numbers on there today. We're not pointing out, some particular solution to the problem of how to value individual welfare. Instead, what I'm trying to do is to get us to start thinking collectively about how we would make these trade-offs in a more principled way and what it would look like. How would that process go, right? The main move that I'm suggesting is to start thinking in terms of a common unit, which might mean borrowing a bunch of tools from the economists. And if we did that, then here's the way this process would go, just focusing narrowly now on one particular kind of case. So imagine we've got a particular biodiversity related intervention. Suppose we're trying to prevent habitat fragmentation, right, in some particular area. And so we're trying to, discourage, a road from being built that would fragment, say, a particular ecosystem or something like that, right? That's the project that we're pursuing. And the way we want to think about that particular biodiversity intervention is we say, well, suppose that we manage to block the thing that we want to block. Suppose that we manage to prevent the bad outcome that we want to prevent. How good would that be, right? What exactly would the benefit of that be? And so then you'll have to price that. But then what you also want to do is discount that by the probability of success. What you want to do is say, look, this is the good thing that we would be able to accomplish if we were successful. But then we want to know, how likely is it that we will actually achieve our goal? Because maybe when we assign a value to this particular biodiversity outcome, it's very high. But tragically, maybe the odds of actually achieving that outcome are extremely low. And so what we say then is, in expectation, in other words, when you multiply the value times the probability of success, or by the probability of success, you get a much reduced value, right? You don't get to claim the whole value, you get to claim some much more modest value. And then what you do is you're going to have some value, or rather disvalue, it's a negative value, for negative welfare impacts if you have to kill a bunch of insects to do, to figure out whether you're actually going to be able to achieve the relevant outcome. We look at that disvalue and we multiply that again by the probability of impacts. But of course, in those cases, the probabilities are going to be very high. We know we're going to do a bunch of collection to achieve whatever the goal is. We know we're going to harm a bunch of individuals to achieve whatever our goal is. And so we can subtract and we can figure out what's going on. And what that would allow us to do here is find out, is that overall value positive or negative? If it's positive, then it's probably, it might well be justified. It might well be the thing to do. And if it's negative, that just means, hey, actually, when we put all of this stuff in a common unit, we found out that the costs to individuals were too high. That is not the way we should pursue the goal. Now, that is very abstract. But if you think about what this means concretely, right, you will see this is the way these kinds of assessments are actually done for lots and lots and lots of other, economic assessments, right? This is just garden variety, standard practice. All we're talking about is extending it to something that has normally not been handled in those terms. We're talking about taking an approach that assigns prices and values to things and says, What happens if we start doing that with respect to insects? And of course, we're not used to thinking this way. We are not used to thinking about the value of harming particular individuals in terms of dollars that have to be compared with the good of, say, promoting biodiversity. I want to resist skepticism about this process, because I think that this is a way that we actually take individuals seriously. When I think about what it means to try to get recognition for animals, to try to get animals taken seriously as beings who deserve some measure of respect in our decision-making, part of that involves counting the costs for them in our overall analyses. Right now, individual welfare just doesn't show up. So when you're counting the costs and the benefits of a biodiversity intervention, of some strategy for improving health of ecosystems or what have you, individual welfare impacts are completely absent. Impacts on humans count, but not impacts on animals. All I'm calling us to do is try to include them, try to figure out how can they show up. And even if the valuations are very low, even if the numbers that we assign don't result in our changing our practice, at least we are now moving toward a picture where animals are visible in these processes. We are now saying very clearly, oh, these impacts count. They are among the things that should be factored into our overall cost-benefit analysis. And so when someone says, well, okay, I want you to factor animals in, individual welfare impacts, as all of those papers do, as I mentioned in the beginning, we can now say, all right, well, at least we have a starting point. We have the beginnings of a framework for assessing what we are doing to these individuals. All right, thanks so much.
Thank you so much, Bob. I think that is a really, really nice way for us to end our conference today. So thank you very, very much for your brilliant perspective, as always. We are running over time a little bit, so I don't think we have time for questions, but would you be happy for anybody in the audience to get in touch with you if they want to kind of chat to you about your framework? Excellent. So if you've got any questions from this audience or online audience, do get in touch with Bob directly. So thank you again. And just to kind of wrap up the conference, because I know a lot of you want to go straight to the top. So just as a big wrap up, I just one thing before we do, we have our student awards. If you want to come up and congratulations, there you go. All right. So yeah, big.
Sorry, say that.
Oh, okay, there's a photo. Do you want to hold that up? Well done. No pressure. Keep smiling. Great. There we go. Well done, Katie. Great. Okay. So yeah, and just to close off, thank you so much. It has been a brilliant day. I don't know about you, but I've got so many new ideas from so many different perspectives, and it's just been brilliant to be in a room with so many different academics. Like, I would have never thought to think about veterinary care for spiders, like old tarantula. I never would have thought about that. And again, I would have never thought about the welfare aspect of dealing with tsetse flies or mosquitoes. You know, that's just something I wouldn't have thought about. And so, you know, thank you so much. And this is what I hoped that we would get from the SIG, to bring together people from such a vast variety of different areas to discuss the same topics. And I'm hoping that this is something that will keep going forward. So as you know, this is the first SIG, hopefully the 1st of May. The plan is for this meeting to happen every other year, but possibly having kind of combined meetings with some of the other SIGs in between. But I'd really like to hear from you guys, both you guys here and also online. You know, where would you know, this is a space for everybody. You know, I want to know what you want to see for the SIG going forward. So please do drop me an email with any kind of feedback about things you want to see or things that would be helpful for what you're doing. And yeah, just a big thank you to everybody. Another round of applause for all of our speakers. I would just like to do a big round of applause for our brilliant student helpers. If you guys could put up your hands. letting us do this SIG. So a big thank you to all of you guys for all of your help and support as well. And there we go. I think that's the end of the first ever insect welfare and ethics SIG. Well done, guys.
Massive thanks to Eleanor for organising as well.
Oh,
Great job.